Iya, informasi kepada Bapak Ibu hadirin, jikalau sudah menyantap coffee break, untuk supaya tidak terlalu tumpah, mungkin piring dan gelasnya bisa diletakkan di luar lagi di meja. Terima kasih. Okay, ladies and gentlemen, we start our next session. Please be seated and ready to hear our speakers. Distinguished ladies and gentlemen, without further ado, let us continue with the next agenda. For the first session, I would like to invite our first moderator, Dr. Yulia Rahmawati from the Research Center for Biosystematic and Evolution to the lead this session. Please welcome Dr. Yulia Rahmawati. Give the applause.
Thank you for the kind introduction. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. My name is Yulia Rahmawati, and I will be the first moderator for our first session today. It is my pleasure to welcome all of you who are present here together with us, and also to all participants that are joining us via Zoom. Welcome to Workshop on Biodiversity, Genetic Resources, and Innovative Bioresources Technology for Future Sustainable Use of Biodiversity. Before we begin our workshop today, I'd like to remind all of you of our agenda. We have 10 distinguished professors, 10 distinguished speakers.
Ibu hadirin, jikalau sudah menyantap coffee break, untuk supaya tidak terlalu tumpah, mungkin piring dan gelasnya bisa diletakkan di luar lagi di meja. Terima kasih.
I'm very sorry for the interruption. So let me continue. So as I said, we have 10 distinguished speakers who are expert in their field, and each speaker will talk for about 20 minutes, and then we'll have 10 minutes of Q&A session. So ladies and gentlemen, I highly encourage all of you to actively engage in the Q&A sessions. Without further ado, let me introduce our first speaker. We have Professor Yoshimasa Tanaka. I'm very sorry. I will read his CV. Please have a seat on my left side over here. Yes, so we have today Professor Yoshimasa Tanaka on my left side. He is currently a professor and the director of Center for Medical Innovation, Nagasaki University, Japan. He has been studying cancer immunotherapy for more than 30 years and has published more than 200 publications in reputable journals. It is our honor to have him join us today in this wonderful occasion. And in today's seminar, he will talk about the drug discovery research and biomedical research in Nagasaki University. Good morning, Professor.
Morning.
All right, so ladies and gentlemen, without further ado, Professor Tanaka, the floor is yours.
Okay. Can I have a slide? Okay, thank you very much for your kind introduction. So today I'm going to be talking about our recent activities for the drug discovery and biomedical research at Nagasaki University. The title is Marine Derived. Okay, thank you very much. So marine-derived microbes and molecules for drug discovery. So first of all, I'd like to thank Andria sensei and Kamitakahara sensei and organizers for giving me an opportunity to share with you our recent activities on drug discovery. So our institute, Center for Medical Innovation, was founded in honor of Dr. Osamu Shimomura, and he graduated from Nagasaki University. And then he discovered GFP, the green fluorescent protein from jellyfish. And then the GFP is frequently used in drug discovery. So we founded this Center for Medical Innovation. And he was awarded Nobel Prize in 2008. And then today's talk consists of 4 parts. The first one is the introduction and monoclonal antibody drugs and marine microbial extracts and nanobody. And then the first part is the introduction. So this slide indicates the code-specific mortality rate in Japan. This axis represents deaths per 100,000 population. And then this axis is the year. And then we don't have the data during the World War II, but before World War II, many Japanese died of pneumonia, gastroenteritis, and tuberculosis, so infectious diseases. But after World War II, malignant neoplasms caused Japanese people's death. So before World War II, many Japanese died of infectious diseases, but the death rate rapidly declined after World War II because of the improvement of nutrition and public health and also development of antibiotics, as you know. And then after World War II, malignant neoplasms caused deaths has been increasing because of the prolongation of longevity. So it is imperative to develop effective treatments for malignancies in Japan right now. So this is the history of the drug development. So the modern drug development started about 130 years ago. And then the first 100 years, and most drugs were small molecule drugs. Most of them are natural products, and then they are developed by serendipity or the development of organic chemistry. So most drugs are analgesics like aspirin and anti-infectives like sulfur drugs and antibiotics like penicillin. And then from the late 1990s, monoclonal antibody So monoclonal antibodies are the main pipelines of the pharmaceutical companies. And then probably the next 10 years or 20 years, the cell-based drugs will be the major drug discovery systems, actually. And then the first 100 years, the most drug targets were receptors that recognize small molecule ligands. So the original ligands are small molecule ligands. And then if we try to develop drugs, that drugs become small molecule because the original ligands are small. So our drugs are also small. And then another target was enzymes inside the cytoplasm that recognize small molecule substrates. So original substrates are very small molecule. So that's why when we try to develop drugs, that drugs become also small molecules. But the problem is these receptors and the enzymes are coded by genes, and we have only 20,000 genes. So the number of targets is limited. So that's why the last 100 years, many pharmaceutical companies try to develop small molecule drugs, but these targets are gone already. And then the next part is the monoclonal antibodies. And then the pharmaceutical companies changed their strategies. So they changed the targets of the drug development. And then that target, the new target is the receptors expressed on the cells, and then the proteinaceous ligands expressed on another cells. So the next target was protein-protein interaction outside of the cells. And then when we try to develop inhibitors for protein-protein interaction, the monoclonal antibodies are the best. So that's why in our system, we use this PD-1, PD-L1, for example. We have to develop monoclonal antibodies specific for PD-1 or specific for PD-L1. And then I had been working with Professor Honjo in Kyoto University. And then, as you know, he was awarded a Nobel Prize 5 years ago. So we are working together. And then until 15 years ago, the standard cancer therapies were surgery and chemotherapy and radiation. But about now, half of the Japanese people suffer from cancer, but half of the patients die from cancer. So we have to develop new cancer drugs. And then the cancer immunotherapy came from the United States, New York. And then this one is Dr. William Cawley. He was a surgeon. And then he developed the Cawley's toxin. One day, one patient came and named Fred Stein, and he had very big tumor, neck tumor. And then accidentally, he was infected with Streptococcus pyogenes. And then after the cure, his tumor was also gone. And then the next patient came, 1891. And then to this patient, the William Cawley injected live Streptococcus pyogenes, and then the tumor shrank down. But it was very dangerous because that Streptococcus pyogenes are alive. And then next patients came who had cancer, and then the William Cawley killed Streptococcus pyogenes to the patient, and then the tumor shrunk down. And then at that time, the cancer immunotherapy started. But the problem is, so many researchers developed the cancer immunotherapy, including pathogen-associated molecular patterns, that is Streptococcus pyogenes, and then cytokines, tumor antigens, and dendritic cells, and tumor-specific T cells, and so on. But the efficacy was very low, something like less than 0.1% or something like that. So most people almost gave up 20 years ago. But then breakthrough came from totally different field. So this one is a programmed cell death using C. elegans. So C. elegans is this kind of worm. And then it started from one cell, fertilized cell. And then the cells divided into 1,090 cells. And then at the certain times, SED3 and SED4 programmed cell death related gene works. And then 131 cells die to give 959 cells to give a form of C. elegans. And then in Professor Honjo's lab, they were interested in programmed cell death in immune system. This one is not immune system. This is the mice in the womb. Their hands actually for food, something like fan. And then just before birth, their hand becomes something like just finger. And then this is a human. Our hands are something like this originally in the womb, but the cells between the fingers die at certain times caused by programmed cell death. And then Professor Honjo's lab, they are interested in programmed cell death phenomena in immune systems. T cells are very important cells in immune systems. And then when we stimulate T cells very strongly, T cells die. And then they found one gene expressed on the activated T cells. So activated T cells express this molecule. So they call PD-1, programmed cell death 1. And then Professor Honjo published the science paper, and then he just stated, maybe PD-1 is involved in programmed cell death, but actually it was wrong. And then after that, Professor Minato's lab, Minato is president of Kyoto University now, and then Professor Honjo's lab worked together, and we analyzed the PD-1 knockout mice. And then when we established the PD-1 knockout biopsy mice, they developed cardiomyopathy, so big hurt. And then we found the deposition of antibodies that specifically recognize troponin I. That means when T cells recognize foreign antigen in the context of M.Si class 1 molecule expressed on an antigen-presenting cells, they recognize antigen-presenting cells. But the problem is the normal cells express self-antigen, and then the T cells recognize self-antigens, and then they kill normal cells. So this is very bad, actually. This causes autoimmune diseases. But if normal cells express PD-L1, that binds to PD-1 and negative signal gets in, and then the T cells no longer kill normal cells. So we can prevent autoimmune diseases. But the problem is, what happens if tumor cells or virus-infected cells or bacterially infected cells express PD-L1? They bind to PD-1, and then T cells no longer kill tumor cells or virus-infected cells or bacteria-infected cells or other infected cells. And then we examined the expression level of PD-1 on ovarian cancer cells. And some patients express a high level of PD-L1, and some patients express low level of PD-L1. And we analyzed the overall survival of the patients. And then when patients who express a high level of PD-L1, the overall survival rate is very low. In 10 years, only 40% patients survived. But the cancer do not express very high level of PD-L1, the patients can survive. So the PD-L1 expression is a prognostic factor. And then we developed anti-PD-L1 monoclonal antibodies that specifically recognize PD-L1, and then that interfere with the interaction between PD-1 and PD-L1. And we established the tumor cells expressing PD-L1 and injected to mice. And then the tumor grew bigger and bigger in the mouse. But when we treated the mice with anti-PD-L1 monoclonal antibodies, the tumor didn't grow very well. So this is the first time we showed that we can cure cancer using anti-PD-L1 monoclonal antibodies. We call PD-1 immune checkpoint inhibitors. And then this is a mechanism underlying the PD-1 ICI. This is the immune checkpoint inhibitor therapy. When tumor cells exist, dendritic cells recognize the existence of tumor cells, and then dendritic cells grab some tumor antigens and they move into lymph nodes. And then they present tumor antigens to naive T cells, and the naive T cells are differentiated into effector T cells, and they kill tumor cells. But if tumor cells express PD-L1 that binds to PD-1, and the effector T cells can no longer kill tumor cells. But if we add anti-PD-L1 monoclonal antibodies, we can interfere with the interaction between PD-1 and PD-L1, and then effector T cells can tumor cells. So this is the mechanism. And then this one is the commercially available drug, anti-PD-1 monoclonal antibody of Opdivo. And then this is the overall survival of lung cancer patients. And then when we treat the lung cancer patients with chemotherapy, only 24% patients survive only 1 year, in 1 year. But when we treat the cancer patients with PD-1 immune checkpoint inhibitors, 42% patients can survive 1 year. So this is a big difference. So that's why PD-1 immune checkpoint inhibitor is called breakthrough of cancer therapy. But 42% is not enough. That is not satisfactory. So we try to increase the efficacy up to 80%. And then we focused on this effector phase. And then when tumor cells are killed by immune effector cells like T cells, the number of effector cells are very important. And then all we have to do is to increase the number of immune effector cells, and then we can kill tumor cells, or we can kill virus infected cells, or we can kill bacteria infected cells. And then we chose IL-18. This is the interleukin 18 to increase the number of effector T cells. So the NK cells, gamma delta T cells, and alpha beta T cells express IL-18 receptors. And then when we add IL-18, that binds to IL-18 receptors, and we can increase the number of NK cells, gamma delta T cells, and alpha beta T cells to kill tumor cells or virus infected cells or bacterial infected cells. And then when we take spleen from the wild mice and then stimulated the T cells, and then T cells proliferate very well. But if we take spleen from IL-18 knockout mice and then stimulate T cells, the T cells do not proliferate very well. But if we add IL-18 to this system, the T cells proliferate very well. So IL-18 is a factor to increase the number of immune effector cells. And then we did experiment. We injected the colon cancer cells to mice, and then we treated the mice with either PBS or PD-L1 monoclonal antibody or PD-L1 combination therapy. And then, as you can see here, after a tumor injection, the mice die, all mice die within 100 days. But if we treated the mice with PD-1 immune checkpoint monotherapy, 50% mice survived. But if we treated the mice with PD-1 immune checkpoint inhibitor combination therapy, all mice survive. So we can cure cancers using the PD-1 immune checkpoint combination therapy. And then this is a CT image of the liver cancer in mice. So about 80% to 90% liver can be cancerous. But when we treated the mice with PD-1 immune checkpoint inhibitor combination therapy, the tumor shrunk down to very small size within only 4 weeks. So this is the strategy. This is the target of the monoclonal antibody. So protein-protein interaction outside of the cells. But this kind of target is also limited. Maybe we have only 1,000 or something like that. So the next target of the drug discovery would be different. So that is the protein-protein interaction inside of the cells. But it's very difficult to inhibit the protein-protein interaction inside of the cells because the monoclonal antibodies are too big. They do not go into the cell membrane. So that's why we have to develop another kinds of drugs. So one is a middle-size inhibitors, and another one is the nanobodies. So we are working on this right now in Nagasaki University. So the third topic is the marine microbial extracts. So this is a chemical space of the drugs. And then this one is the medicines on the market. The chemical space is very big, actually, something like this. But the natural products, the chemical space is almost the same as medicines on the back market. So that means many medicines are derived from natural compounds. Maybe 40% of medicines are from natural compounds. We usually use the synthetic compounds for drug discovery, but the chemical space of synthetic compounds are very small. So totally different from the natural compounds. So we have to use natural compounds for drug development. And then we decided to use the marine microbes to develop the middle-sized molecule drugs. So this is a map of the Nagasaki prefecture. the southwest part of Japan. So Nagasaki Prefecture and many islands and gulfs and bays. And then we collected many marine microbes. And then we sampled a lot of marine microbes. And then this is the Tsushima Island. This is a very beautiful place. And then we collected sea urchin and crabs, shrimps, sea cucumber and sea anemone and oyster or something like that. And we isolated marine microbes. And then we grew microbes and then extracted the middle-sized molecules. And then we purified these middle-sized marine microbe-derived compounds and then screen many drugs. And then the final talk is the nanobody. This one, nanobody is the heavy chain only monoclonal antibodies derived from alpaca, the camel animals, and also from shark. And then we decided to use shark. But before that, I'll introduce the camel. And then now the most monoclonal antibodies are humanized monoclonal antibodies. And then probably the next generation, next 10 years, many pharmaceutical companies develop single chain antibody and bispecific antibodies. And then probably after 10 years, many people work on nanobodies derived from camel or shark. And then this is the structure of human IgG. So Y-shaped monoclonal antibody. And then the next generation antibody is a single chain antibody. And then further, the bispecific antibodies. But the nanobody came from totally different place. This is the University of Brussels. And then the students are working on monoclonal antibodies. But at that time, the HIV infection was a big problem in 1980s. So students did not work on HIV patient samples or something like that. So they decided to use animals. But ordinary animals are not so interesting. And then, incidentally, they have a serum from camel. And then they analyzed the monoclonal antibodies from camel. And then the camel antibody structure is a little bit different from our antibodies. So they have the ordinary antibody have heavy chains and light chains, and heavy chains and light chains. But the camelate heavy chain only antibody consists of only heavy chain. And then we just take this part. This one is a variable region of the heavy chain only antibody. So we call this one a nanobody. And then the camelid animals derived from something like Arizona or something like that in the United States. And then they went out to North and Alaska, Siberia, and then China and Africa. So they are dromedary and Bactrian camels. And some animals went south. And then they became llama, vicuña, alpaca, and guanaco. All of them have heavy chain only antibody. And then we call, as I told you, this part only, we just take out this part. This one is called nanobody. So molecular weight is about 1/10 compared to the human monoclonal antibody. And then this is the characteristics of the nanobodies. And this nanobody contains long CDR3 regions. CDR3 is a complementary determining region 3, and that specifically interacts with antigens. And then this nanobody is physico-chemically stable. If you increase the temperature up to 70 or 80, that also works. And then affinity is very high, and we can produce nanobodies in E. coli. So that's why we can prepare very inexpensive monoclonal antibody preparation. And then the gene modification is very easy. And then to use camelid animals, it's difficult in Japan. So that's why we decided to use sharks for the preparation of nanobodies. The shark heavy chain only antibody is something like this. So very similar to camelid antibody. And then they also have a variable region, and we can just take out this division. And then we call this one also nanobody, that's specifically recognized protein. And then we, in Nagasaki University, we have Department of Fishery. So we have the ship, something like this, and to catch the shark. And then we have shark farming system in the Department of Fishery. And then this is the shark farming system. And then we keep, this is the bait of the shark. This is a squid. And then we can have Nurse shark and also Nanuka shark or something. And then we can, this shark is very small, so we can just hold this one. And then we can take blood, something like this. So it's very easy to take blood, something like this. And then this is me. And then I can deal with this small shark. And then I can take just blood from the shark, something like this. And then we can just read the genes, and then we can read these genes, and we can synthesize the gene for the variable region, and we can prepare nanobodies. And then this, the molecular weight of this nanobody is just 1/10 compared to a humanized antibody. So we can use this nanobody to interfere with the protein-protein interaction inside of the cells. So this is all. Thank you very much for your attention.
Thank you very much, Professor Tanaka. That was such an interesting talk about his work in cancer immunotherapy and especially his recent work on nanobodies development from shark. Now, please have a seat. Thank you so much. Now we will have a 10 minutes discussions since we have audience here together with us and also audience via Zooms. So for those of you who are here, please raise your hand and state your name before asking your questions. And to those who are joining us via Zoom, please type your question on the chat box. All right, now I'll take some questions from the floors. Please do not hesitate to ask questions, as it is such a rare opportunity to interact with the experts. Yes, please. Can we have the mic?
Okay, thank you, Professor Tanaka, for the excellent presentation. I just, nanobody is not really my expertise, but I would like to know, like, what's the advantageous nanobody compared to like the regular monoclonal antibody?
Monoclonal antibody. Your question is the difference between the monoclonal antibodies and nanobody?
Yeah, and the advantages, like what's the why you choose nanobody and not like the traditional monoclonal antibody?
Yeah, okay. So ordinary antibodies. So in 1980s, many Japanese people, especially the Tohoku University people, they are working on monoclonal antibodies. At that time, they couldn't produce the human antibodies. But 1990s, American scientists developed a method to produce the humanized antibody in mice, actually. So they exchanged the immune system of mice to the human systems, and then they can produce the humanized antibodies in mice. But the problem is that monoclonal antibodies are very successful, and then maybe more than $70 billion sales in worldwide. But the problem is the target of the monoclonal antibodies are only protein-protein interaction out of the cells, because the molecular weight is 150 kilodalton. So they can interfere with the protein-protein interaction outside of the cells. But if we focus on the protein-protein interaction inside of the cells, that monoclonal antibody has to go into the cells. But the monoclonal antibodies are too big. So that's why we need the very small monoclonal antibodies. But it's very difficult to prepare monoclonal antibodies, because the ordinary human monoclonal antibodies, the B region consists of two domains. So one variable domain from heavy chain and one variable domain from light chain. And then that protein has to interact with each other. So if so, on the surface of the protein, there are hydrophobic regions. And that is the cause of the instability of the protein. So that's why we cannot use the human variable regions. But in case of camelid nanobody, and then the shark nanobodies, they do not have hydrophobic region on the surface of the molecule. So that's why the nanobody is very stable. So if you increase the temperature up to 70, 80, the protein is still stable. So, and then they can be expressed in E. coli. So the price is very low. So maybe if so, maybe you can use the nanobody drugs in the maybe ASEAN countries, African countries, or South American countries also. Right now, the humanized antibody is very expensive because they are produced in CHO cells, Chinese hamster ovary cells. In order to grow CHO cells, the media are very expensive, but the E. coli the culture media is LB media, so that is not so expensive. So that's why we can produce the nanobodies inexpensively. Okay.
Thank you, Sensei.
Thank you very much. Yes, we have a second question over here. Can we have the mic, please?
Thank you for the opportunity, moderator, and thank you, Sensei, for your nice presentations. My name is Naro from Research Center for Pharmaceutical Ingredients and Traditional Medicine, BRIN. So, just a quick question, Sensei, about your presentation. What I got from your presentation is that there are two receptors expressed in different cells in the cancer. The first receptor is the PD-1 that is expressed on the surface of T cells. And the other receptor is the PD-L1 that is expressed in the tumor cells. So when the PD-L1 attached to PD-1, then the effectivity of T cell to kill tumor cells is decreasing, right? And according to your presentations, what I got is you and your team or other researchers in the world are developing the antibody that recognize the PD-L1, right? To inhibit the binding of PD-L1 to PD-1. So I just want to know, Sensei, has it been evaluated the effectivity between inhibiting the PD-L1 and inhibiting the PD-1? Which one is more effective in order to kill or to suppress the number of tumor cells in the body? Thank you so much, Sensei. Thank you so much.
And so actually the PD-1 has two ligands that is called PD-L1 and also PD-L2. So if you use the antibody against PD-1, you can inhibit the interaction between PD-1 plus PD-L1 and also PD-1 plus PD-L2. So, and then, but if you use anti-PD-L1 monoclonal antibody, you can inhibit the interaction between PD-1 and PD-L1 only, not PD-1 plus PD-L2. So if so, maybe it's better to use the PD-1 antibody. But the problem is the PD-L2 is usually expressed on dendritic cells. That is typical antigen presenting cells. So if so, it may cause autoimmune diseases. So that is the problem. The efficacy of PD-1 antibody is good, but it may cause autoimmune diseases. And the efficacy of PD-L1 monoclonal antibody, the efficacy is a little bit less, but it doesn't cause autoimmune diseases. So that is a big question, but I prefer monoclonal antibody against PD-L1 because it doesn't cause autoimmune diseases. The efficacy is a little bit less, but it's okay. But and then also, if we add IL-18 to the therapy, that increased the efficacy of anti-PD-L1 monoclonal antibodies. So that's why we are now using anti-PD-L1 plus IL-18 as a PD-1 immune checkpoint combination therapy.
Thank you. Thank you so much, Sensei.
Thank you very much. We still have a couple of minutes for few more questions. So please, audience, if you have questions, raise your hand. All right. If there is no question from audience, allow me to ask a very simple question. So your topic is really interesting because it is something new for me as I'm not from the field of cancer immunotherapy. So you talk about nanobodies, how it is very stable since it doesn't have a hydrophobic site, and it can be produced in large scale in the E. coli. You only touch a brief view on that. So I'm wondering, how far are you in the research of this nanobodies production? And have you tested for the side effect of this side effect or the cytotoxicity of these nanobodies? Thank you.
So actually, there are already the couple nanobodies on the market already, but maybe now many researchers are working on nanobodies. So maybe 10 years later or 15 years later, many nanobody drugs will be on the market. And then when it comes to toxicity, and then the nanobody protein is totally different from human monoclonal antibodies. So maybe the nanobody can be recognized by human immune systems, and then it causes some cytotoxicity. But we can mutate the nanobodies and shark nanobodies in order to escape from the human immune systems. That is also possible.
Right. So in your research, you also mutate the shark nanobodies to imitate like human nanobodies.
Yeah, that's true.
Right. Okay. Thank you so much, Professor Yoshimasa Tanaka. I'd like to call for one more. Okay. So there is no more question, and I'm afraid to say that this is the end of our first session for today's discussion. But please, to all participants, stay because we still have 9 more experts to talk about their topic. On behalf of the committee, I'd like to thank Professor Tanaka for his great talk and very informative presentation on cancer immunotherapy. Ladies and gentlemen, please give a big round of applause.
Thank you very much.
As the moderator, I'd like to apologize for any mistakes I made during this session. I'd like to pass the mic back to the MC. Thank you very much, and wassalamualaikum warahmatullahi wabarakatuh.
Thank you to our speaker and our moderator. Please join me giving a warm round of applause again to the speaker and the moderator. Okay, distinguished participant present here and online participant. Let's us move for the next speaker. But I would like to invite Dr. Taufik Purnanugraha from the Research Center of Applied Zoology to lead the session. Please welcome Dr. Nugraha. Time is yours. Give applause.
Good morning, ladies and gentlemen. So now we proceed to the second talk. As the moderator for this talk, I would like to welcome Professor Gono Semiadi to the stage. Please welcome. It is my honor to introduce Professor Dr. Gono Semiadi, an esteemed zoology scientist with over 3 decades of research experience. Prof. Semiadi is currently affiliated with the Research Center for Applied Zoology at the National Research Innovation Agency. He is also a renowned expert in mammals ecology, taxonomy, and conservation biology. He has played pivotal roles in international conservation efforts, serving as national focal point for Indonesia CITES, SAPSTA, UNCBD, and IPBES. Professor Semiadi impactful contributions to this seminar. He will present us with insight about database of Indonesian mammals hair for forensic. Professor Gono Semiadi, the floor is yours. You have 20 minutes for presentations.
Okay. Good morning. Good morning, ladies and gentlemen. and thank you for the opportunity to present my speech. And my topic is about how the basic science can contribute to the tackling the illegal of the trade, in which one approach is through the identification of hair as the one of the evidence in the prosecution. The illegal wildlife trade is an organization transnational crime and has been a problem that has been difficult to address for many years in many countries. This crime is a profitable business that target for life or death the skin and body part of the wild animals of the Asian region is mostly as the mostly is affected by this illegal trade. The Asian region is a center for the illegal wildlife trade, and the positions of the countries among the Asian countries mostly can be as a source of the animals. Also, the country can be positioning as the transit routes. And finally, some of the country in Asian region can be as the destination for any illegal wildlife trade. In Indonesia, as one of the Southeast Asia largest supply of the various type of the exotic animals within the illegal wildlife trade, Indonesia has approximately 707 species of mammals, which representing almost 12% of the world mammals. Also, Indonesia harbor almost close to 1,600 species of the bird, and among others for the fresh fish, from the amphibian and reptiles, and also from the mollusks and the crustacea. Not to mention as well from species from the marine area. A survey of the illegal wildlife trade in Indonesia shows that the avian constitute as the top species being illegally traded, and followed by the mammals and the last of the reptiles and amphibians. If you look at the mammal species distributions as part of the illegal wildlife trade, primate is the top priority to be poached for the illegal trade, and followed by the carnivores. The large number of the entrance and exit point within the coastline of the country is a problematic itself for the country to protect of the any illegal transit of any Indonesian species. The key to combating the wildlife crime is almost the success of the investigations and the effectiveness of the law enforcement. And that needs support from the technology to uncover the evidence. The applications of the science in the crime investigation, known as the forensic science, has been developed through capacity building in law enforcement in Indonesia for quite some time, either within the Ministry of the Environment of the Forestry, also in the police or the attorney level. Apart from that, a distributions of a handout or pictorial of the species has been distributed for quite some time in many of the point of the exit within the Indonesian coastline harbor. The obstacle is when the nature of the illegal species has been converted or processed to other form of the specimen. Morphology, anatomy, and molecular approach have been the standard procedure for the forensic investigation for quite some time. Morphology character can be used to identify quite precisely for the species, to some degree for the age estimations, and also to some degree for the approximate of the distributions. Whereas the anatomy and pathology, apart from the identifying approximate of the species that have been targeted, also can be identified for the age and the approximate dispersion apart from the cause of the death and the pathology. With the molecular, as nowadays mostly can be said as a standard goal among the forensics works for the illegal wildlife. This molecular can be also precisely identified to the species, sometimes to the status of the populations apart from the approximately distributions, and also to identify to a certain degree whether this is the species is coming from the wild or from the captive origin. However, those three science or approach, it sometimes cannot support what is needed to identify a certain part of the body of the wildlife animal. That comes when a microautonomy or histology approach is needed. Among of the evidence that usually being used for opening up for the evidence is using the hairs. And we already know that hairs of the body part have been a good source for the key identifications. However, in Indonesia, this approach has not been well implemented, neither has been well developed as part of the forensic science. The use of the hair to solve the crime has been proven in many or several cases, mostly in the European area. Several manuals of the hair identification has been published and has been used as the protocol for gaining a good evidence. However, to my knowledge, most of the majority of those handouts is very minimum for the discussing from the tropical species. As a national depository, BRIN, or used to be the LIPI, stores thousands of the specimen of the Indonesian species and can be the primary source for the developing hair identification of each species. Museum Zoologicum Bogoriensis, which is established almost 150 years ago, deposited almost 2 million of the Indonesian specimen across all of the Indonesian species and still growing for its depository number. This is a vast resource for us to develop a database of the Indonesian wildlife hairs, in particular if we're discussing about a pure of the animal contradict to the hybrid species, which already happened quite sometimes in Indonesia to certain numbers of the species, of the wildlife species. For the past 4 years, we have conducted a research on several hairs coming from the Cervidae, Bovidae, Suidae, and Felidae taxon groups, where from the Cervidae, we do have a lot of problem in identifying at least coming from the 2 species, which is the Rusa unicolor and the Rusa timorensis. Whereas from the Bovidae, Banteng, where in a certain part is really isolated within the national park, has been a main target due to their close performance with their local, what we so-called the Bali cattle, which is coming from the Banteng as the main source for their genetic pool. Within the Suidae, it is a problem quite sometimes in quite numbers, a mixing between the Suidae origin to the regular domesticated meat has been done just for the sake of the profit. Whereas the Felidae, as we already know, with the Sumatran tiger, it's always been as the main target for the poaching, with almost all of the part of the Sumatran tiger has a high value of the commercial. So this is for the first years that we are focusing, and we are still continuing to finish it up from those 4 taxa. But perhaps in the future, we are going also to come across the avian group, mostly to the Passeridae, whereas the bird, as we already mentioned in earlier, that avian group put as the first numbers within the illegal trade within the Indonesia. So when we are conducting of the sampling of the hares, since mostly at the moment we are focusing on the collective of the scientific specimen, we do have some limitations for the hair collections, not too many parts that we can use. So far, we are only able to collect subsample from the dorsocranial and the dorsocaudal section of the body. And within, in the Suidae, in the certain numbers, we do also collecting a specific hairs coming from the bear or from the face sections. In the futures, we do hope that we can start collecting samples from the wild or from the zoo collection, where we can collect a lot of, quite many numbers, the positions of the hairs, a part of the hair type that we are able to collect. So some characteristic, if we look to the result from the cuticle scales of the Indonesian species, if you look at into the, on the left side, it is within the Suidae, and the far end, it is from the Rusa. It can show that to a certain degree that the cuticle scales, if you look at precisely under the microscope, it can be used and we can find quite number of distinct character. However, within the Suidae, that character is not as strong as what we find in the within the Cervidae. Similarly, and if you will look to the medulla, it shows also a not quite strong within the Suscrova, but it is a quite strong character which is shows within the Cervidae group or the Rusa. And last, from the cross sections, a specific character is quite distinct among the species within the Suidae as well as within the Rusa. And this character, it is a character that we might further develop for deciding as the solid character when we are coming for the evidence or identifying the species. So within the limited number of the specimen and the species and the body part or the section of the hair, we were able also to construct the identification key from the hairs of those 4 animal groups. And we are certain hope that this key can be used as well in the future as a baseline for identifying the species, either from the perspective of the basic science, and also for the implementation through the forensic science. Several characters we are still looking, and we are developing a more simple guiding for using hairs as the key identifications. And in the futures, once we have done having a quite number of the baseline data of the hair characteristic, eventually for the practicality in the field, either used by the police or anyone else, we are trying to develop an artificial inseminations using our database that can be implemented within the simplest application, then at least can help the what we so called the parataxonom or the non-taxonomy people to at least identify as an early stage before an in-depth prosecution or in-depth of the identification of the species in the laboratory can be conducted. The future research is, as I mentioned, is establishing a database of the wildlife health, in particular, identifying what is the best character between the pure and the hybrid species to those species that has been either deliberately cross-hybrid, as it happened within the Felidae, the small Felidae, and also in the large mammal, in particular, like in the Bovidae, and also in a certain degree in the Anoa, between the mountain Anoa and the lowland Anoa in Sulawesi. Secondly, as far as we are trying to make a coordination with the Ministry of the Forest and Environmental for how this Indonesian database can be implemented and a part of the system within their platform. Secondly, we are trying to establish the Indonesian wildlife database, of course, based on the stable isotope character, as within the Indonesia, that any confiscated specimen, the regulations is requested to have a precise locality, the origins of that confiscated species. So we are trying to look if the stable isotope can play a role in that positions and integrate it with our work for within the identification of the species through the hands. Lastly, improving our artificial intelligence applications that we might be much more usable for much broader community. I thank to the JASTIP. Throughout our study, JASTIP has also contributed to our study in a certain part of the facility, at least for the providing the chemical for our study. Thank you very much.
Thank you very much, Professor Gono. Professor Gono has shared a talk on groundbreaking initiative in analyzing wildlife hair and develop essential metadata and key identifications for develop from Indonesian species. And this will be bridging from basic science to applicable tools, assisting law enforcement efforts to tackle illegal wildlife trades. Now we will proceed to the discussion sessions. We have around 10 minutes for discussions. I'll encourage you all to actively engage with our speakers. Before we proceed, I also kindly request that participants would like to ask to mention their name and affiliations for better context. This will help our speakers address our inquiries more effectively. So is there anyone that would like to have questions? Okay, please. Microphone. Please kindly mention name and affiliations. Thank you.
Thank you very much. My name is Dharmayanti from Research Center for Genetic Engineering. For Prof. Gono, it is very impressive research in your area. I just want to know how effective is like hair for the forensic method in your research right now and in the future for molecular forensics method for combating the illegal wildlife trade. For example, can this method differentiate between like mammal in the wildlife in the museum or in the zoo, or it is a hybrid? For example, no, not hybrid. I mean, what is it? domesticated wildlife or something like that. Can your method differentiate between that states of animal? Thank you very much.
Okay, Prof. Gono, you could proceed to answer the questions.
Okay, thank you, Wendy. The approach from the hares, we are trying to contribute for the simplifications in giving the answer of the evidence, because under the regulations, the police can only withhold the persons only to a certain hours, which is 48 hours before it is decided whether it has to be released or can be temporarily withheld for the further in-depth version. So yes, as I mentioned, molecular has been a standard, the gold standard. However, there is some quote-unquote complaint about the timing of getting the result. That is the first one. And the secondly, sometimes is the process of the samples, the evidence. Usually there is some complaint about it is not quite good, takes quite some time because of this has been rotten, deteriorated. And if you look at the situation, that won't happen within the hares. It's almost as a stable positions, only becoming a fractions, either because they regurgitated or has been the cut. So that is the approach, and that has been some argument why for the forensics coming to the, when it is the evidence, there is an opportunity to get the hares, they will go to the hares as well. And secondly, as I mentioned, because our, even though it's been 4 years, our samples is at the moment is still limited from the specimen collections. We do have some restrictions on utilizing the samples because honestly, if it's taken from the specimen collection, it is invasive work. We have to take out or cut the hairs from the specimen, which is at the moment, it is quite difficult to get some permit. But the next one, yes, we do try to distinguish the strong character between the pure and the hybrid, and between the pure and the domesticated. It is actually our aiming, but at the moment we are not able to collect quite numbers. We started with the Bovidae, but it only comes from the Banteng first, that is the much easier, coming with the Bali cattle and the pure Banteng. And the next one is perhaps to the Felidae, which is because if you look at to the Facebook, quite very numbers, they deliberately hybridize this wild small Felidae carnivore to the domesticated cat and sell as the high price, quote unquote, a good new domesticated hybrid. So we are hoping the contribution of the hares is quite significant in the level for the prosecutions. Thank you.
Thank you for the answer, Prof. Gono. Any other questions from the audience? And also welcome from audience in Zoom also to ask question in the chat box. Okay, for the second question, Prof.
Thank you very much, Kono Sensei. I have a question about the specimens. And you mentioned that there is some limited number of specimens. But for stable isotope technique, I think you need to have the specimen from some localities, not for only one animal from one locality. And that is important for comparing the natural and domesticated animals. So do you have any kinds of strategy that you are getting samples from natural area?
The question is, if it is coming to the zoological specimens, the first question is how big the sample that you are needed. When it comes just for a minute quantity, even though we take it from the several parts, it's much easier for getting the permission compared when I work with the hairs. And secondly, I don't know what is the interference with the stable isotope due to the processing of the museum specimens. Because at the moment we are working from the pre-colonial period, which is during the time they are using quite numbers with the arsenic chemical. And so that is the positions of need something to be discussed when we want to go to the stable isotope work. And secondly, we are still looking for the candidate to do that as well, as the proposal. Thank you.
Thank you very much.
Thank you very much. Is there any other questions?
Sorry, Mr. From the Zoom, have the raised hand.
Okay. Anyone raising hand?
I'm sorry, this is.
I don't see anyone raise hand, so. Okay. Yes.
Please. Mr. Bramantio.
Okay. So we have a question from Zoom audience. Yeah.
Okay, Mr. Bramantio. Do you hear me? Please open your mic.
Okay. So we have one question from Mr. Bramantio. So while we're waiting for the question, is he able to connect yet? Not yet. Oh. As Prof. Gono mentioned before, like for the first questions, like the things about this method is like we try to find some like more simple methods, like to identify in a very short time, because with molecular is like the golden standard, but of course sometimes we just need to know if this animal is protected or not protected. So it's a very short time windows. Either like the prosecutor could detain the one that have these animals or these samples or not. So that is very crucial. And if we only depending on molecular identifications, it will takes at least 1 week or even more, even if we go for sequencing. Any more question?
No.
Okay. So we could still have like one more questions if anybody will be interested to ask. Otherwise, we thank you. We will end these sessions. So we would like to thank you to Professor Gono Semiadi for sharing with us. Let's give him a warm applause. And I'll give the time back to the MC. Thank you very much.
Once again, and thank you to our speaker and our moderator. Thank you, Dr. Nugraha, Taufik Purnanugraha. So we are continue agenda. For the next speaker, I would like to invite Dr. Iksan Guswendrivo from the Research Center of Applied Zoology to lead this session. Please welcome. Dr. Iksan, time is yours.
Thank you very much, Mr. MC. So very good afternoon, everyone. These sessions I will lead before we listen to Professor Kenji Umemura presentations. I would like to introduce for all the audience that Professor Dr. Kenji Umemura is a research professor from Kyoto University, a renowned professor in wood science and technology. Professor Umemura research focus on groundbreaking advancements in bio-based adhesive adhesion systems and the sustainable use of agricultural waste in wood-based materials. So without further ado, we will listen to the third speaker that will deliver by Professor Kenji Umemura with the title Biodiversity for Sustainable Product Wood-Based Materials. So please, Sensei, time is yours.
Yeah, thank you very much. So I am Kenji Umemura, the Research Institute for the Sustainable Human Sphere, Kyoto University. So first of all, it is my great pleasure to have this opportunity. So today, I'd like to talk about the biodiversity for the sustainable production of wood-based materials. So before my presentation, I'd like to introduce myself and my laboratory briefly. I'm a professor of the laboratory of sustainable materials. And my research topics, there are three topics. First is the exploration of the wood adhesion technology. And the second is the development of the wood-based materials. And the third is the degradation and the durability of the wood adhesive. And my laboratory aims to develop the new wood-based materials harmonized with both and the global and the regional environment by leveraging their characteristic of the lignocellulosic biomass. In addition, the integrated project among the interdisciplinary fields are being carried out to confirm the sustainability and the production and the potentiality of the lignocellulose biomass. Okay, so this is the global forest distribution, and here is the boreal forest, and this is the temperate, and this is the subtropical forest, and here this is the tropical forest, and this is the subtropical forest. And as you know, the global forest area in the 2020 is estimated about 4.06 billion hectares, which is the 31% of the total land areas. And 45% of world forests are the tropical domains. This is account for the 45%. And followed by the boreal 27%. This is 27%, and the temperate 16%, and the subtropical is the 11%. But this is the change of the global forest area from 1990 to 2020. So as it, this is the change of the global forest area, and the forest area declined gradually, and 178 million hectares have been lost. And the declined area is equivalent to the 4.7 times of the land area of Japan. And the rate of the forest loss slow recent years compared to the from 1990 to 2000 here. And this is due to the forest preservation and the increase of the plantation forest. But the forest area is expected to continue to decrease, considering the deforestation and the climate change. On the other hand, This is the global trend in the wood removal from 1961 to 2021. So this is the roundwood. So the volume of the roundwood increased 1.6 times over the 60 years. And as you know, the roundwood is being used in two ways. One is the wood fuels, the other one is industrial roundwood. And the production is the wood fuel in 2021. Here is about 2 million. And the industrial roundwood is also the 2 million. So this means that the utilization of wood has accounted for the 50% of the wood fuels and the 50% of the industrial roundwood. And now the production of the wood-based panel is added in this figure. So this is the wood-based panel here. So the wood-based panel include the plywood, particle board, OSB, and fiberboard. And the production of wood-based panel increased gradually like this. And 10% of the roundwood is used for the wood-based panels. And about 20% of the industrial roundwood is used for the wood-based panels. Anyhow, the utilization of wood and wood-based panels increased year by year. Considering the global population growth and economic development in the future, the utilization of roundwood, including wood-based panel, is expected to increase. But you know, the forest provide the multifunction, not only the material production function, but also these functions. For example, the biodiversity conservation function, the global environmental preservation function, blah, 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 like this. And the decrease in the forest area means the loss of these functions. And therefore, the forest preservation will become more and more important in the future. And considering this situation, we need to think the utilization of unutilized lignocellulose resources as raw materials for wood-based materials. For example, this is the global cereals production from 1990 to 2020 here. So this is the total production. So you mean the production increased linearly, and considering the population growth and the economic development in the future, the production will increase. This is the Asian regions. The Asian region also increased linearly, and here is about account for the 48%, about 50% in the total. And the cereals include the wheat, rice. Oh, sorry. The cereals include the wheat, rice, corn, sorghum. And these crops, cereals, generate the agricultural waste, such as the straw and stalk and the bagasse. And you know, the open burning of these waste is prohibited due to the air pollution and global warming. Therefore, we need to deal with these waste. So to summarize these things, the forest area will continue to decrease due to the deforestation and the climate change. But the production of wood-based materials, including the panel, will increase in the future. So this reason is due to the population growth and the economic development. Therefore, we need to investigate the alternative raw material for wood-based materials. On the other hand, the cereals production will increase. With this, the agricultural waste will also increase. In particular, the huge amount of the waste are generated in the Asian regions. So, but the open burning of these waste is prohibited due to the climate change. Therefore, the utilization of the agricultural waste is important issue. So we need to investigate the utilization of the agricultural waste as a raw material for wood-based materials. Basically, the wood-based material consists of the wood element and adhesive. So when we develop the sustainable wood-based materials, we have to think the sustainability of the resources of wood and the element. So this means that the sustainable and renewable of raw material are very important. And as to the wood element, the agricultural waste is one of the promising resources. And as to the adhesive, the synthetic resin is generally used. And these synthetic resin adhesive are basically synthesized with the chemicals derived from the fossil resources. So here there are many types of the synthetic resin. For example, the formaldehyde-based resin and the PMDI, epoxy resin, and so on. And this table shows the raw material of synthetic resin adhesive. So this is the raw material, and this is hydrocarbon, and this is resources. So as you can see that here, there are many types of the raw material. For example, the polyvinyl acetate and polyvinyl alcohol and the vinyl acetate and the styrene and so on. And these chemicals are derived from ethylene, and here from propylene, and here is the butan-butadiene cut, and here this is from the aromatic compound. And these hydrocarbons are derived from the naphtha. And in case of the formaldehyde here, this is from the natural gas. And this naphtha and the natural gas are derived from the fossil resources. So almost raw material of synthetic resin adhesive are derived from fossil resources. So considering concern about global warming and the potential shortage of the fossil resources, so we have to reduce the consumption of the synthetic resin adhesives. And then, so we need to replace the synthetic resin adhesive with the bio-based adhesive as much as possible. And this is the bio-based wood adhesive. The bio-based wood adhesives are classified into 5 groups. This is the saccharide-based, protein-based, aromatic-based, oil-based, and others. And in the saccharide-based adhesive, the starch is used as raw materials. And in here, the soy protein and the aromatic animal glue and the casein is used as raw material. And here, the lignin and the tannin is used. And here, the natural gas, the vegetable oil and the bio oil from the lignocellulose are used as a raw material. And here, the natural rubber also used. And many researchers have been researching this bio-based adhesive for the practical use. However, there are many challenges. So because this is the problem with the biosubstances as a raw materials. So basically, sometimes the biosubstances have the chemically stable structure and high molecular weight and wide molecular distribution. and non-uniform and complexity chemical structure. Therefore, when we develop the bio-based adhesive using the biosubstances, the obtained adhesive have the sometimes low reactivity and insufficient polymerization and severe curing condition. In addition, this is the requirement of the biosubstances for the practical applications. So when we use the biosubstances, we need to think the price and the quality and the amount and the availability. So this means that biosubstances have the variety, locality, and the seasonability. So therefore, we need to take account for these things when developing the bio-based adhesive. And when we develop the bio-based adhesive, there are 4 research directions. The first is the utilization of the natural substances in conventional synthetic resin. For example, the chemical structure of the tannin and the lignin are similar to the phenol. Therefore, instead of phenol, the tannin and lignin are used when synthesize the PF resin. And the PF resin containing the tannin or lignin are synthesized. And the second is the development of the adhesion performance by chemical modifications. So this means that the starch soy protein itself have not the bonding property. So to develop or to enhance the bonding property, some chemical modification is needed. And the third is the improvement of the adhesion performance by adding synthetic compounds. So in this case, the addition of the isolate resin is a popular method to improve or to enhance the bonding property of the bio-based adhesive. And the fourth is the combination of the 1, 2, 3. So in this case, first, we synthesize the PF resin, including the lignin or tannins. After that, so to enhance or to improve the bonding property, the addition of the synthetic resin is added. But in those research directions, so there are some problem. Here, this is these research directions. There are some problem. Here's the complicated preparation and high dependence on the fossil resources and low adhesiveness, especially low water resistance. Therefore, when we develop the new bio-based adhesive, we need to think the simple preparation and low dependence on the fossil resources and the high adhesiveness. Anyhow, when we develop the sustainable wood-based material, we should think the utilization of unutilized lignocellulose and bio-based adhesive. This means that the keyword of the biodiversity is very important in the wood-based material in the future. And this is research topics on the development of the sustainable wood-based material. So our laboratory have been investigated the wood-based material using agricultural waste as raw materials. So here, this is the particle board using the oil palm trunk, inner part of oil palm trunk, and the ammonium dihydrophosphate and the sucrose. And here, this is the particle board using the sorghum bagasse and the citric acid and sucrose. And here is the particle board using the sorghum bagasse and the kaizen adhesive. So now I'd like to introduce this research topic briefly. So the oil palm plantation area in the worldwide is 21.2 million hectares. and oil palm is replanted every 24 to 26 years, and a significant amount of the oil palm trunk is generated. And generally here, the inner part of the OPT become a waste because of its insufficient properties such as the low strength and the low durability due to the much amount of the parenchyma cells. So, but in our laboratory, we purposely used the inner part of OPT as a raw material for particle board. And in this case, we use the ammonium dihydrogen phosphate as additives, and also we use the sucrose as the adhesive. But first, we try to use the ADP only to manufacture the particle board. So first, we prepare the ADP water solutions, and this solution was sprayed onto the particles. Then after making the mat was hot pressed. So we obtained the particle board like this. This is the mechanical and the physical property of the binder's particle board using the ADP. So here, this is the ADP amount, and this is MOR, and this is MOE. And here, this is without using the ADP. In this case, the MOR value is about 5 megapascal. But when we use the ADP of 10% here, the MOR value about 9 megapascal here. So the bending property of the particle board was improved significantly by the addition of the 10% of the ADP. And figure 2 shows the dimensional stability of the binder's particle board using ADP. So in case of the without using the ADP here, the thickness swelling about the 50%. But when we use the ADP about the 10% here, the thickness swelling about 5.9%. So the particle board with the ADP addition of the 10% addition of ADP had the excellent dimensional stability. So we investigate the reason for the improvement of the property by addition of the ADP. So it is known that OPT contained many saccharides such as sucrose, and based on our previous research, it was clarified that the sucrose changed to the water insoluble substance under the condition of the heating and existence of the ADP. Therefore, the saccharides, including the sucrose, would change to water insoluble component during pressing. And the water insoluble component would act as adhesive, and the OPT binder's particle board would develop their good properties. So it can be said that this adhesion system utilized the chemical component of the raw materials. According to this finding, the saccharide is a very important role for the improvement of the properties. Therefore, we saw that the addition of sucrose in addition of ADP would be effective for further improvement of the board properties. In other words, the synergistic effect of the sucrose addition is expected to bring the further improvement of board properties. So, first we prepared the ADP and the sucrose water solution, and this solution was sprayed on the particles, and we manufactured a particle board. This is the effect of the sucrose and the ADP ratio on the bending property of the particle board. So for example, this is the weight ratio. For example, 80 to 20 means the sucrose 80% and the ADP 20%. So this MOR value is about the 60 megapascal. Compared to the binder's board here, the the bending property increased significantly. And this is the board bonded with the synthetic resin adhesive. This is the PMDI. This is the this is a PF resin, and this is PMDI. So as you can see that the compared to the board bonded with the synthetic resin adhesive, the MOR value was a little inferior to that of the board here, but the MOE value is similar to the board with synthetic resin adhesive like this. And this is the effect of the scrolls and the ADP ratio of the thickness swelling and the water absorption of the particle board. So here, the 80 to 20, the board thickness swelling about 10%. So this value is a very good value compared to the binderless board like this. And compared to the board bonded with the synthetic resin here, this value was the similar values. Therefore, the addition of the sucrose was very effective to enhance the board properties. So this is the summarize of the particle board using the inner part of oil palm trunk and the ADP and sucrose. The addition of ADP was effective for the improvement of the physical and the mechanical property of the OPT particle board. And the saccharide would change to the water insoluble component under the condition of heating and the existence of ADP. And the insoluble component would act as an adhesive. And the mechanical property of OPT particle board was significantly improved by the addition of sucrose along with ADP. Consequently, the particle board using the inner part of OPT could be manufactured by environmentally friendly method. And this is the final slide. The utilization of agricultural waste will be important as a raw material for wood-based materials. This means that the biodiversity of the raw material used for the wood-based material will become important. And the adhesion technology that utilize the bioresources will be essential technology in the future. This means that the biodiversity will become important for the adhesion technology. Therefore, the keyword of the biodiversity seems to be the important more and more in the field of wood-based materials. With this, I conclude my presentation. Thank you very much for your kind attention.
Thank you very much, Professor Umemura. So now we will go through the discussion sessions because we are running off the times. Maybe I will just take one question from the floor. If any questions from the audience. Yes, please. Dr. Gege.
Thank you for the moderator for the time. Sensei konnichiwa. Thank you. Glad to see you here in Indonesia now. I am very happy today because thanks for your interesting presentation, and it's also I'm a little bit have a conducting together research with you at this research project, and during that time. And then I'm very surprising because your invention here, by using the bioadhesive material, is have a good durability against the termite and also wood decay fungi. And my question going to if the sucrose or any saccharide able have a potential to use as a bioadhesive, have you considered about the waste lignocellulose mass, like oil palm trunk or sugar gas itself, have a lot of saccharide inside the mass. So can we optimize that compound in the lignocellulose mass, or can we use another more eco-friendly other than the synthetic sucrose? How about, because we have a lot of high field of the sugarcane plantation in Indonesia, or oil palm plantation in here, and they only like using the no one optimized about the sucrose that live in the lignocellulose mass. Thank you for the.
Yes, thank you very much. So you said there are many type of the plant, and including the saccharide. Then so I introduced our research using the inner part of oil palm trunk, but we also investigated the utilization of the bagasse and so on. And I think our aim to use the chemical component in the inside of the raw material. Then so we focused on the saccharide, especially sucrose. Then so also we investigated the curing mechanism of the sucrose itself. Then so the sucrose can cure by addition of the some acid compounds. That's why so we also use the raw materials containing the saccharide. Yeah.
One quick continue question. So is it better to extract the sucrose first from the lignocellulose mass?
Sorry?
So is it better to extract first the sucrose from the lignocellulose waste?
Yeah, but when we manufacture the wood-based materials, so we have to use the raw material as the.
Yes,
that's why.
true.
Yeah.
Yes, true. Okay, thank you, Sensei, for the answer.
All right, thank you very much for the questions. And Sensei, there are two more questions from the Zoom, so I will try to read. First question is from Professor Yulianto. I will try to read. Konnichiwa, Umemura Sensei. I'm sorry I could not attend the event. My question is, why binderless board do not develop well when compared to the development of wood-based materials using adhesive? Okay, I will try to repeat again. Why binderless board do not develop well when compared to the development of wood-based materials using adhesive?
Not well.
Yeah, he says not well.
Yeah, because the bondability is not so good. I mean of course we can get the some the particle board to having the some properties to some degree, but of course the how to say, the without adding anything, so the bondability is not so good. That's why property is not so.
All right, so it will be not so very good for the result. Okay. Hopefully, Prof, you can listen to the answers. And then the last one is from Nurfianto. The question is, what is a good management model in agroforestry when food crops such as soybean, rice, corn are planted in resin or bio-based wood adhesive producing tree stands, so that the bioadhesive industry between food crops and industrial tree is well established? So it's kind of how to maintain both industry together. Yeah, between the agroforestries and also for the industrial tree, it will be established together.
I'm sorry. Familiar with that.
All right. Okay, maybe Mr. Nurfianto is not Prof. Umemura's field, so maybe we will try to find a good question, a good answer for your questions. I think this is the end for the Professor Umemura Sensei's sessions. Let's give a big round of applause for Professor Kenji Umemura.
Thank you very much.
Thank you very much, Professor Umemura. Right. So the next speakers, so I will still be the moderator for the next sessions. So without prolong these sessions, I will continue to the fourth presenter that will deliver by Miss Fanny Clara Ardianti. So before we listen to her presentations, let me give a brief introduction for her background. Miss Fanny is a researcher from Research Center for Applied Biology, sharing her expertise in environmental engineering. Ms. Fanny's research focus on innovative solutions for textile waste water treatment and microbiological approach in shrimp aquaculture. So please join me in welcoming Ms. Fanny to give her insightful presentations with the title about advancing textile industry wastewater treatment, immobilizations technique, metabolic pathway, and reapplication using tropical whiteroot fungi. So Ms. Fanny, time is yours.
Okay, thank you very much, Dr. Rivo, for the nice introductions. And Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone, distinguished professors, also guests and colleagues. So my name is Fanny Clara Ardianti. I'm from Research Center for Applied Microbiology BRIN. And first of all, I would like to thank JASTIP scheme and also the committee for the opportunity. And on behalf of Dr. Dede Heri Yulianto, so I would like to extend his apologies for not being here today since he's on abroad now. He's not in Indonesia. So I would like to give my best to deliver what he would like to share with us today. So before I begin, I would like to share this. Yeah, actually, this is the video, but yeah, well, I would like to draw your attention to take a look at these images. So on the left side, you can see the distressing scene actually of the untreated textile wastewater and depicted by the lifeless fish. So there is actually the fish here, and it's the actual experiment we did when back in one of the city in Indonesia. And yeah, so it's a vivid representations of these challenges of the untreated textile wastewater. So it's not only resistant, but also it's a toxic, mutagenic, and also carcinogenic. So it's posed with a very serious threats. But if we take a look and shift our gaze to the right image, we can see the clean treated wastewater. So symbolized by the lively fish. So this is actually should be the videos, but it's alive. So I hope you believe this is a very lively fish. So this stark contrast embodies the essence of our journey today. So we are on a mission to turn the left side to the right side. So we would like to turn the environmental challenges that we are addressing today to find and seeking the sustainable solutions. So I would like to present our research activities. Actually, this has been supported also by JASTIP. Since 2015, it's about the advanced textile industry wastewater treatment, immobilization techniques, metabolic pathway, and rail application by using tropical whiteroot fungi. So this is actually the, unfortunately, the real pictures that our team took in since 2015, also in some areas in Indonesia. So you can see, we can still spot this unauthorized, actually, textile wastewater dump into the water bodies. And globally, it is estimated around more than 200 1,000 tons of dyestuffs as actually produced globally and annually. And we can imagine that by the increasing population, and I believe we demand the clothes, and also if you are familiar with the trend of fast fashion, so yeah, we can now buy more and more clothes in a cheaper price. So yeah, you can imagine how big it is for a demand for a textile. But talking about dye wastewater, it's not only from textile industries, but also from printing. We can talk about food and paper industry, for example. So basically, there are many type of dyes, but yes, it is originally designed to resistant. So it's because we would like to have long-term use of these dyes. But yeah, unfortunately, like for example, azo dyes as 60% of the most utilized dyes in the textile industry is resistant to aerobic wastewater treatment. So in, like, for example, in Indonesia, as we know, we still have the conventional wastewater treatment. So for example, just the pond with the aerator. So by that, the azo dyes, with its stability, and also it's very hard to break down the azo bonds of the dyes. So it's very resistant and it poses the environmental problems. So yeah, take a look at the textile dye wastewater. So as I mentioned, it is not only toxic, but also mutagenic and carcinogenic. And the textile dye wastewater, it's not only contains about the dyes, but it also has many other hazardous compounds such as salts, acids, and also heavy metals. So I myself witnessed the skin of the skin irritations to some people actually, and especially for the people living nearby the river, because, and also close by the textile industries, since they are using this water for daily purpose like washing and bathing and even drinking. So it's very sad to know that there are still some cases in Indonesia for this health effects. So this is very serious threats, not even for the human health, but also for the environmental risk, especially for the aquatic habitats. So as we know, there are three actual principal techniques for the textile wastewater treatment. For example, the first method is a physical method that we know, the adsorption method. So where the dyes can undergo the porous solid surface of the adsorbent, and it will absorb into the intensive surface of these adsorbents. While for example, the membrane filtration, it will act as the selective barrier where the dyes will be removed by this membrane, and so there will be the decolorization of the textile wastewater. And if we are talking about the next method, for the chemical method, the very famous one is the coagulation and flocculation. So coagulation involves a process of destabilized particles in the solutions, and where the flocculation is the process where these destabilized particles make a contact and make a larger particulates, and then it will sediment, like there are still precipitates that will settle to the bottom. And if we're talking about another process, for example, the advanced oxidation process, so it involves the free hydroxyl radicals, so where the dyes react with that, and it can be also created with several methods. For example, the Fenton process here, and then we also have the ozonation, where the ozone reacts with the dyes and then decomposed as the oxidized products and oxygen. But yes, so the next method, what we call the biological method, or we are familiar with bioremediation. So in this method, we can utilize the microorganisms such as bacteria, fungi, algae, yeast, and also their enzymes to actually the principal process here is to biotransformations of the toxic dye wastewater to the non-toxic compounds. And yes, of course, for this conventional treatment, there are some limitations of each of them. For example, like if we are talking about chemical methods and like coagulation, and also actually including the membrane filtrations, it offers a very rapid process. So it's very fast, it's very simple, it's very straightforward. But unfortunately, it's relatively economically not feasible for like a small and medium enterprise like we have today, mostly in Indonesia. And also it's relatively requires high energy maintenance costs. And also as for coagulation and flocculation, we have still a sludge generation. So we still have a ways that we need to think about of the solution of this sludge. So I don't know if it's work. Okay, but yeah. So this is actually some videos of Dr. Dedes and our team actually going to Pekalongan City. So Pekalongan City is located in Java Island, and it's famous for called to be a batik city. So they produce a lot of batik in a small and medium business. And and this project actually we also collaborate with one of the Japanese companies producing polyglue. So this polyglue is a bioflocculant which contain from the fermented soybeans. So we try several, like just a quick demonstrations to them, like how if we applied this polyglue. And as you can see, this is the untreated wastewater, and this one is the treated one by the polyglue. And this is just a small scale wastewater treatment that we built just to provide the process of this polyglue. So it will not need many times, like it's just in a short time, maybe 1-2 minutes, and we add polyglue, the bioflocculant here, and then we are doing here the filtration, like simple filtration process. And then after that, we can see this clean water, but as you see, there are this floating sludge created by the coagulation and flocculation process. So these collaborations is actually, they would like to know like if this floating sludge is hazardous or not. But once again, it's the concern, it's still producing this sludge. So therefore, we need to have the efficient treatment methods. So if we take a look from the techniques, one of the methods which is promising is adsorption methods, whether it's chemisorptions or by chemical reactions or physical sorptions. So yes, it's over easy, simple, flexible treatments. Nowadays, like researcher is going to study about the many type of adsorbents. For example, many researchers explore what kind of the low cost adsorbents. Usually they are from the natural material and also from the waste. So since we would like to utilize the waste into something more valuable, so like for example the agricultural and industrial waste, we also research about carbon-based nanomaterials. In our project, I think we try these HNT materials, hollow nanotube. And then also the biosorbent. So it's the microorganism itself as the adsorbent. And it's very, it has the maximum removal efficiency since we try to utilize their adsorption capacity at the maximum. Okay. It's okay. So the next promising method is microbial the biological methods. So for example, when we are using the microbes, so it's actually simple as well, and it is famous for the it can degrade the toxic compounds into non-toxic compounds compared to the physical and chemical method. But unfortunately, like it has a relatively slow process, like for example, 6 to 30 days. So therefore, we need one of the solutions that we need to find the super microbes, right? Like really, they can degrade into faster process, or like we can also try to the immobilization method that I will talk about. And then if we compare to enzymatic biodegradation, then it will be faster, but it's very challenging for low cost production. So yeah, therefore we also need to finding novel enzymes that we would like to try now. We would like to utilize the enzymatic engineering, like to find maybe from a recombinant microbes or from that, so we can have very novel enzymes, and also we can use the immobilization method. So here we combine the biological and physical methods, what we called by the immobilization. So it's basically the entrapment or the attachment of these microorganisms or their enzymes into the adsorbents, like a carrier or a solid. And then what we would like to achieve is that it has the maximum adsorbent capacity. So we try to take the advantages of these physical and biological methods. So this is actually the roadmap of our research activities. Actually, yes, it has been supported by JASTIP SIM since 2015. So in 2015 until 2017 or 18, we have been very active to have an explorations from Aceh to Merauke to explore, like doing the isolation, screening, identification of potential fungi, at least like 100 samples, but maybe now we have 17 potential isolates of fungi, especially the white rot fungi. And also after that, we've been doing the selection of the fungi to degrade the dyes. Here, like a synthetic dye wastewater, whether it's in the solid or liquid medium. And maybe in 2018 and 19, we try to develop the enzymatic degradation. So we try to extract the enzymes of the fungi, especially the lacase. And we also have the patent for this enzyme productions of lacase to degrade the dyes, the synthetic dyes. And also after that, we in 2019 to 2020, I guess, we try to explore like the immobilization technique on mycelial and enzyme on biomaterials. So we're trying to find what carriers is the best for this immobilization. And then after that, since 2021 until now, we are trying to develop the pilot application and implementation, and we hope also we can apply this in real wastewater treatment. So the key technology here is actually we would like to combine the protein, what we call protein biomaterials, so it can offers the more stability and reusability. Yes, and we are doing this project with Professor Takashi Watanabe from Kyoto University, Japan, and also since 2019, we collaborate with Kyungpook National University, Korea, with Professor Sungkwan Kim, and also University Science Malaysia. So we're doing a very big projects with everything. And then I think it's not working. Sorry. Next. Yeah. Okay. So our research of interest, actually, we mainly focus on the white rot fungi. So this WRF, it can degrade dyes because it can secrete the ligninolytic enzymes. So it can consist, for example, the manganese peroxidase, lignin peroxidase, and also lacase. But in our research group, we try to focus on these lacase enzymes. So actually, the first technology is because of that the white rot fungi can degrade lignin, but since it has a characteristic to be a non-specific, so it can degrade not only lignin but also other various xenobiotic compounds, like including dyes. So the main technology that our research group developed is the fungal immobilization in light expanded clay aggregate. So I think if you are familiar with this carrier, so you can find it for like when you are gardening for the plant. So it has the very porous characteristics, so that we utilize this leca as a carrier, and we're doing the steam activation process to make a bigger porous of inside the surface of the leca, and then we wash it, and then after that we try to make the liquid medium of our white rot fungi, and then it's homogenized, and also after that it was added to this leca. And by doing the incubations and of course optimization process, and then we can have this mycoleca. So this mycoleca actually have been registered a patent, and this is just an example of some images of the inside infrastructure of the mycoleca, and you can see the mycelia here. It's actually not only outside the surface, but also it's entrapped to the inside of the leca. So we are doing the various decolorizations. So first for synthetic dyes, many of dyes here, and then also the textile and batik dye wastewater. So it's a real wastewater that we try. And you can see the color itself, it's decolorized from here to here. And this one also from one of the textile industry in Jababeka industry here. And yes, so we also do the toxicity assessment. So here, yeah, actually there are some reference here, but you cannot see. So in 2021, we published a paper in a collaboration with Kyungpook National University. So they help us also to do this detoxification on human dermal fibroblast cells, or HDF. So as you can see, from 0 hour to 24 hours, the cell viability is increasing. And also we're doing the phytotoxicity and the toxicity assay on Artemia salina. So after that, we also doing the metabolic pathways. So we here actually with the KNU, we use the UPLC high resolution mass spectrometry. So we would like to analyze what is the biodegradation pathway of these dyes. For example, here is the anthraquinone dyes degradations, and also for the azo dyes degradations. But here, so yeah, we use the UPLC FTI CRMS, and then we try to analyze the biodegradation pathway. But here we are adding the molecular docking study. So to have a simulation is actually like we would like to find a reason why one of the dyes cannot be easily degrade. For example, like here for the azo dyes of reactive black 5 dye, we can see the peak of the spectrum disappear, and this one is the the pathway. Also the same as the acid blue 113 dye here, but for the AO7 or acid orange 7 dye, it's very hard to degrade. So by this analysis of the metabolites and molecular docking self, so the reasons is that we find a weak affinity of these dyes, and also the absence of the interaction of the residual active enzyme sites with this as the azo bonds of the acid orange 7. And yeah, these are our 2 papers. This one is from the Journal of Hazardous Materials 2021, and this year also we published a paper in Chemosphere. So yeah, if you would like to take a look, we can see. And yeah, for the time,
yes,
okay, okay, I will just very quickly. So yeah, as I said about the reusability, we are doing the test for the Pekalongan printing and Purwokerto batik. So this can be used for 19 cycles, and we also monitor the LECAS activities. So the result is that there's still some activities even for the last cycles. And this one is just a simple application, since the batik small batik industry would like to know the technology. So here actually before and after just 24 hours, since this is utilize the microorganisms, it requires longer process actually, and also in the factors of the wastewater itself, like 3 to 5 meter cubic per day. So it requires here is 3 weeks. But yeah, we monitor the pH, COD, and the colorizations, and also these enzyme activities. Yeah, and this one is just another application in real wastewater, and this is actually the ongoing project that I lead as well, for fungal immobilization on Calnus. So this Calnus carrier, so we're talking about the plastic carrier here, which is actually has been developed since 19 80s, but it's rarely explored with the fungi. So we're trying to develop in this career. And yeah, we would like to also thank the collaborators from the industries and our team. And yeah, thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh. Thank you very much, Miss Penny, for the very good and big project, I guess, because their project is to collaborate with a lot of industries and as well as many universities, not from Indonesia, but some from Korea and Malaysia, if I'm not mistaken. Okay, because we have only limited time, I will give only one question from the floor. If there is any questions.
Maybe everyone would like to have lunch break.
Maybe. Because it's a very big project for the environments, I guess. So I think from my perspectives, from my perspective, what do you think about the future for the real applications? Because maybe you will face the real problem when you try to apply it in the real situations. What do you think about it?
Yes, thank you very much for the questions, Dr. Rivo. So yeah, we still have a lot of homeworks in these real applications, especially with the enzymes. So now we're still trying to develop this microorganisms immobilization technique. And what usually the users here, like the industries in the small and medium business, they're really concerned about the cost.
Yes.
So the cost is very important. So that's why we're trying to develop the process where it's optimize the medium or the carrier, so it can be very affordable for the users.
Okay, because maybe it's not just the cost. We know that like the last sentence you say is that if you use fungal, you need times to see the results. So I think it will be another problems that you face when you try it in the real conditions.
Yes.
I think that's all for today's sessions. Let's give a big applause for Miss Fanny. So before I give the mic to the MC, I can give you all the participants the summarize from these two presenters today. From Professor Kenji Umemura that discussed the crucial link between biodiversity and sustainable wood-based materials products, that Professor Umemura highlight that the potentials of agriculture waste as a key for resources and proposing bio-based alternative to synthetic resin adhesives. And for Ms. Fanny, she addressing the environmental impact of the textile dyes wastewater is urgent, and her presentation highlight groundbreaking solutions by using combinations between biological and physical method. So it will enhance efficiency, reusability, and stability, making a transformative leap in sustainable wastewater treatment. So I think that's all for me. I will give the mic to MC. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you to our speaker and our moderator. Please join me giving a warm applause again with the speaker and moderator. Now, but to all participants, allow me to once again invite Dr. Taufik Purnamanugraha again to lead the last presentation in this morning session. Time is all yours.
Thank you, MC. Good day, everyone. So I hope you could still hold for another 30 minutes before lunch. For now, for the last session for today, I would like to invite our fifth speaker, Professor Dr. Ichiro Tayasu, to the stage. I would like to invite Professor Dr. Ichiro Tayasu to the stage. I'm sorry. So, okay. So let me introduce Professor Ichiro. is a distinguished environmental researcher and deputy director of General at the Research Institute for Humanity and Nature. His talk exploring environmental dynamics through multiple isoscape hopefully will give up insight into the revolutionary environmental traceability methodology. Professor Ichiro, the floor is yours. You have 20 minutes for presentations. Thank you.
Yeah, thank you very much for presentation, for invitation. And I'm sorry it's time for lunch, but I must speak. And thank you very much for inviting me for this nice workshop today. And I thank you very much for organizing committee for preparation. And also, I'm sorry for my voice is not very good, and sometimes I may have a strong cough because of some kind of asthma symptom. Sorry. Okay, I will present my talk. And the title is Multiple Isoscapes on Environmental Research. I'm Ichiro Tayasu, and the co-authors with Shin Kicho, who is the colleague of the center of our institute. Today's talk is consists of these topics. And firstly, I must introduce my institute. This is not Kyoto University and a different institute, and especially for studying environmental issues in Kyoto City also. And it is located in Kyoto City, but it's here, but Research Institute and Kyoto University main campus, and this is RISH and Center for Ecological Research, is a little bit different place. And only this institute organizing environmental isotope study, and this project is started from the Professor Wada and Professor Nakano, who is the specialist of stable isotope ratios in light elements and also heavy elements. And we have several instruments to analyze stable isotope technique, and some are for elemental analysis, and these are for isotope ratios of light elements, and these are for heavy elements. And in our institute, we can analyze these isotope ratios of elements, and from light elements to heavy elements. Some are already ready for the analysis, and some are just under preparation. And we hope to analyze many elements to analyze, and that's usable for environmental issues. And first, I will introduce how to interpret isotopic information. And the important point is source and isotopic fractionation in these elements. And first, I need to explain the expression of isotope ratios. And isotope ratios is a kind of ratio from the, for example, for hydrogen, the light isotope of elements and heavy isotope of elements. But usually expressed in delta notation here, because the ratio is fluctuates only a little bit. So we use this kind of expression compared with standard and sample. So first I need to express about the source and isotopic fractionation, because for the study of isotope ratios, the ratio itself is not a problem, and we need to compare the isotope ratio of something and the different substances. So usually we need to know the isotope ratios of substrate, but or living organisms depends on the source. In the case of plants, source is a kind of CO2 or some nutrients. And for animals, source is food. So we need food for lunch, but you need to hear a little moment of my presentation. But the important point is that a source value is 1 point. And another is isotopic fractionation. What is the change of isotope ratios from the source and materials, or food and living organisms? And for these issues, we need to know, it is required to know the spatial and temporal information of an isotope ratio of the source. And the next is for isotopic fractionation. For these issues, we need to know the process-based information via fractionation factor, which is studied in various aspects, but we need to know this kind of information before analyzing isotope ratios of material or living organisms. And this method is sometimes called the stable isotope fingerprint, in which the stable isotope ratio of each element contains its information of origin and pathways of reactions. For example, you know this figures, this one. This is a very famous plot of measured by started by killing in Hawaii. That is an increase of CO2, and now it's around 420 ppm on the Earth, and at the same time, isotope ratios of CO2 is decreasing because of the human anthropogenic impact by fossil fuel burning. And because fossil fuel has a lower value of carbon-13, so that value, if in coincidence with increasing CO2 by decreasing isotope ratios of delta-13 of CO2. And this is the source for plants. So this value is also important for plants. And this information is isotope ratios of plants, which is published in many years ago, but still valid. And if you look at the proportions of isotope ratios, this from -10 to -30, -40, and the distribution is quite different between C3 plants and C4 plants and the CAM plants. Mostly the C3 plants are forest plants, and the C4 plants is some kind of herbaceous plants in tropics. And the CAM is also a different type, but you see the different distribution is happening along the delta-13 values. Although the delta-13 values of CO2 is here. So that is the difference. It came from the isotopic fractionation during the photosynthetic process. You mean that's the delta-13 of plants, delta-13 C of CO2 minus some kind of delta, capital delta values. And these values has been studied and calculated using this kind of equation. And there are some factors, but the important point is CI divided by CA, which is located here. And this value is smaller in drier condition, which means depends on the stomata openness. That means if the stomata is closed in the case of drier conditions, that means this ratio is smaller. So in that case, this value is smaller, and this value is smaller, and the minus value is smaller. So it means from this original value CO2 to moving this direction smaller. So using these equations, you see the variation of C3 plants during this area. It came from the function of precipitation. In the case of very dry conditions, the value is more positive, but in the wet areas, the delta 13 value is more negative. So this is a kind of isotopic fractionation during the photosynthesis. But importantly, we have the different characteristics. This part is very negative values, and this can be found in source effect. That is, the CO2 came from soil respiration. In the case of soil respiration, has much abundance. In that case, the forest floor plants fix carbon CO2 during photosynthesis. But this is the delta 13 value of CO2 in that area is different from worldwide CO2 delta 13 values. So we have the distribution of carbon 13 values that are dependent on source effect plus isotopic fluctuation during photosynthesis. So if you look at carbon 13 values that came from the two processes, that is quite important factors. And also if you look at nitrogen isotope ratios, that is the plants use nitrogen from some nutrients. Nutrients can usually considered as for the nitrate, ammonia, or organic nitrogen, or some other factors. So the plants utilize these nutrients from the natural system that depends on, for example, nitrates. Nitrates has a wide distributions. Came from if the plants capture nutrient from nitrification, the value is more negative. And if the nutrients, the plants utilize nitrogen from nitrogen fixation, that is close to zero by definition. And this part, Professor Koba will explain more details about these processes. But in any cases, plants nitrogen isotope ratios of plants depend on nutrient sources, and we need to know the source of nitrogen in that area. And for animals, what determines isotope ratios? As I explained, carbon isotope ratios of plants depends on photosynthetic pathways and, of course, source of CO2. And if animals eat these plants, carbon isotope ratios change only a little. However, nitrogen isotope ratio increases about 3 or 4 per mil per trophic level. So if the animal eats these two sources, two foods, the value is intermediate between the two. So if you look at the natural systems, there is a food web structure expressed here, and the trophic position is calculated by nitrogen isotope ratios, and food sources are expressed by carbon isotope ratios. However, as I mentioned, different forests have different plants, and these values are different, and also the different areas of ecosystem, they use different source of nitrogen. So it's difficult to explain if you have only one samples here, which area we have samples, because it depends on these base factors. So that is a very important point for using stable isotopes for natural systems. For example, this is the study of human isotope ratios of human depending on age. The first two panels above the result of analysis by more than 4,000 years. We can analyze isotope ratio of prehistorical humans by analyzing nitrogen and carbon isotope ratios of bones. And you see the difference in among Japanese. Japan has very different in nitrogen and carbon isotope ratios from north to south because of they utilized the local foods. So we see the very wide difference between them. So if we analyze the isotope ratios of human of that age, we can identify where the human were there. However, for example, this is around Edo era, so for 16th to 18th centuries. In that time, mostly the mainland was the similar isotope ratios. Instead, Hokkaido has different values. That means they still have a different habit from mainland Japan. And nowadays, the isotope ratios are similar because we buy foods by supermarkets. So supermarket is widely distributed in Japan, and they just sell everything from Japan, also US or China or any places. So the value is similar. So they are all human, but the isotope ratio is different, depends on their feeding habits. So that kind of information is quite important for analyzing stable isotope ratios of wild animals. So like isotopic fractionation is, as I explained, some kind of discrimination factors and also nutrient uptake, and also trophic change is quite important for distinguishing isotope ratios. And also decomposition process change the organic matter isotope ratios of organic matter. Sorry. So next I explained by isoscape study. Isoscape is the word of landscape plus, isotope plus landscapes. And that means distribution of isotope ratios. It's explained in Japan's isoscape, but in anywhere we have the isoscapes of different isotopes. And also we, as I explained, we offer the stable isotope measurement of various elements. So we suppose that multi-isoscapes will help for identifying multi-source problems. So this is like a concept of multi-isoscapes that we use many layers of isotope map to identify the distribution, the origin identification of materials or living organisms in many cases. This is called forensics as well. And for different elements shows the different type of information, which I will not explain in details, but different isotope ratios have different meaning. So I explain some type of isoscapes, and this is a delta to H values, which is hydrogen isotope ratios, and this can be obtained from this site, and this is just gathered many isotope ratios from the world, and for the world scale, it has been obtained, and these can be this can be distinguished between these areas and you see that there are some kind of effects, and especially that the information has been used by American continent, North American continent, because the trend is very well studied, and from heavier, from higher isotope ratios here and lower isotope ratios here. So in that case, there are many studies used for identification of the places, especially for this monarch butterfly, which flies from Florida to North America. And this is region of Asian regions. And also there are some kind of difference between them. And however, it's not well written in Japan. And in that case, we studied isotope ratios of water. Now, we have a difference between the north to south. And if you look at the Tohoku region, the more detailed study has been done. And in that case, we have this strong difference between the areas. So if you look at more details, you have the different isotope ratios. And from Indonesia, Professor Ichiro, who is Kumamoto University, has in collaboration with some Indonesian people and measured isotope ratios of water. And I have never met him, but you see these person from, you can see, but some ITR Indonesia. I don't know this institute, but they have information about isotope ratio distribution in that areas. And another type of information is strontium isotope ratios. I don't have enough time, so I don't explain, but this is a basic idea of using strontium isotope ratios, because from the this is the came from the ratio of rubidium to strontium, and strontium is basically all of the materials is stable isotopes, but rubidium is radioisotopes, and some rubidium decays depends on the time, and if the rubidium has much amount, the change of isotope ratios of strontium that is calculated by 87 divided by 86 increases, depends on time. So that is used for earth science, but we don't need to discussion about age, because that half time is 488 million years. So we don't care about this one here. So, but we see the distribution is like this. So using this information, we can distinguish the place of origin of these information. And for China, Korea, Japan, we have more detailed isoscapes here, but we don't know about Indonesian distribution. So if you have this information, we can see more details about this forensics purposes. This is the areas like this. We have more information about this one. So conclusions. So we have the stable isotopes of light elements and heavy elements, and these elements, each element has each information. And if you have a map, you can see the the identification of sources. But importantly, as I explained, for example, carbon nitrogen isotope ratio is not distributed like a map, but it depends on each nutrient cycling. So we need to know the isoscape type of study, but it depends on the character of each element. Thank you very much.
Thank you very much, Professor. So it's a really interesting talk about the stable isotope ratio, and I think it's also a great potential to also study to make this isoscape in Indonesia, which you say that's still like not many data is still available for Indonesia. So I think it's for the future direction. I think if you could have this detailed isoscape of Indonesian stable isotope, that would be one thing to start to also to support like things like the one that Prof. Gono has done, and also like Bruli will be presented about the use of this stable isotope for forensic approach or for traceability of plants and fauna. So we still have like 5 minutes. I think we could still have like time for one questions from the audience. Is there any question from the audience in the room? Or from the Zoom, is there any question from the Zoom audience? Okay. Okay, you could have. Can we have a microphone?
Thank you for your talk, and we now realize how important it is to have the isoscapes for the application of stable isotopes like isotope forensics or something like that. So what is the most difficult point to establish the isoscapes? For example, when they start to developing the map of isoscapes in Indonesia, what is the most difficult point?
Thank you for nice. We also have a isoscape in Japan, but still difficult to apply in many cases. Some part is because, for example, for strontium isotope ratio is dependent on base rock distribution. However, this isotope ratio of rock, it's not equal to isotope ratios of strontium in plants or animals, because they use only available strontium, not for the rock itself. So the source of, for example, that this is forest, and just the plant is staying here and using these sources. These strontium isotope ratios came from the ground floor of water. So they utilize this one. Although the rock itself here is different rock type, but this water source came from this area. So in that case, for the distribution of geological study has been done, and rock type has been identified, but strontium isotope ratio of available strontium is different. This is the kind of scientific problems. And another is database, and we would like to set up the isotope database. And however, if the database doesn't cover everything, that is the problem. So in US, they started to use establish the ISOBank, that is a repository of isotope data of various informations. And if the database is well established, we can use this database. Although we don't have the database in Japan, but isotope data should be unified in globally. Otherwise, it's if the local, only the local information is not useful, but for the establishment of world level information is quite important. And we collaborate with this ISOMAS group to put the information was data in one repository. And in that case, we utilize this information in Indonesia too, because only aspect ratio of Indonesia is not enough, because we need to contact with other countries around this area, because some, I don't know, but if some animals is imported from other countries, that value should be identified, and we know the isotope ratios of the distribution of isotope ratios. I think this one is a kind of practical issue.
Okay, any more questions? So probably just one question for me. Like couple years ago, like United States like asking Indonesia for sample from honey, because it's like one of the most fraud food consumption in United States, because like most of these honeys like came from different countries, like say from China, and it's like laundry, and it's like labeled like from Indonesia. So the United government asked like for the sample from like all honey for all the regions, then they're going to do this stable isotope analysis. But at that time we still like have like concerns about the data and about the use of the data. Do you think is there's any other implications of like if we like kind of like giving our like isotope data like for the whole country like to other countries? Is there any other implications besides for the study?
Yeah, that depends on the type of fraud. And for example, for the famous fraud for honey is adding C4 plants, C4 sugarcane plants. So in that case, it's easier for identifying C4 and C3 plants, which I explained. That difference is very large. So it's easy to identify it. For example, in our case, someone brought honey from some country. I'm sorry, I haven't explained, but in that case, when I measure this one, it's completely C4 plants, and before that is not a mixture, but a pure C4 honey. So in that case, it's easier to identify. But for different information, it is difficult. If they use C3 sugar from northern parts, that will be difficult to identify. So it depends on what type of contamination of fraud cases.
Okay, thank you very much. So that's probably like a wrap for this session. So thank you very much for your presentation. That give us a warm applause for Professor Ichiro. And probably this is like the end of this afternoon sessions, and we're going to continue to lunch. So I will give the floor back to the MC. Thank you very much.
Thank you to our speaker and our moderator in this morning session. Give applause again to moderator and our speaker. So, excellencies, ladies and gentlemen, before we move on the afternoon session, we will now have a lunch break. Food stalls are set up in the lobby for all participants, and for our estimate guests and speaker, we are prepared the VIP room. The ladies' prayer room is located on this floor, while the male prayer room is located in the basement of this building. Please remember to come back to this room at 1:30 p.m. I also would like to remind you again that the poster presentation will be conducted during this lunch break. Please be present and enjoy your lunch. Please take your lunch time. Thank you. We'll be back again. Ladies and gentlemen, the agenda workshop to begin in just 5 minutes again. And to all participants, please enter the room and please be seated. Hello everyone, welcome back in this workshop on biodiversity, genetic resources, and innovative bioresources technology for future sustainable use of biodiversity. Now we will start our afternoon session of this workshop. I would like to invite Dr. Naraputra from the Research Center for Pharmaceutical Ingredients and Traditional Medicine to lead the discussion. Please welcome Dr. Naraputra.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. So I'm very happy that you're still here to enjoy the seminars and also to have a very strong endurance. And we'll be having for another half day
until the closing of these JASTIP seminars. Today we'll be having the 6 sessions that will be delivered by Dr. Shoji Maehara or Maehara Sensei. But before I pass the mic to Maehara Sensei, I will be delivering some information about Maehara Sensei. He was studying bachelor. He was studying at Fukuyama University to pursue his bachelor's degree until doctoral degree from 1996 to 2013. And Dr. Maehara has interest in the endophytic fungi. Some of the publications discussing about, for example, in 2023, he and his team published an article about anti-SARS-CoV-2 agents in Artemisia endophytic fungi and their abundance in Artemisia vulgaris tissue. And also about the environmental differences between Japan and Indonesia that provide endophytic diversity associated with Artemisia plant and variety of artemisinin derivatives in microbial conversions. And today, Dr. Maehara will be delivering a talk about the innovative use of endophytes against infectious diseases, the case of Artemisia plants. Without having any delay, I will pass the mic and session to Dr. Maehara. Sensei, onegaishimasu.
Thank you very much. Introduce for me. And today invited this workshop. Thank you very much to Kamitakahara sensei and Andria-san, and now already retired Kanzaki sensei. Okay. Now my topic is that he introduced, and I focused endophytic fungus. Many researchers today coming endophytic microbe research, so they know about endophyte. So after that, our group focusing medicinal plant and also focusing infectious disease. Okay. This work is collaborated with Professor Koichi Watashi. He is absent today, but he is core member of WP3 of JASTIP. And together with Dr. Shogo Nakajima, they are doing SARS-CoV-2 assay. It is important section of our working. And in Indonesian Artemisia plant, doing Professor Andria and his staffs. This is collaborated work. It is not possible for my laboratory alone. Okay. Introduce about endophytic fungus, endophyte. This is endophytic fungus fiber of Aspergillus. Endophytic microbes can live in plant cells without injuring their host. Virtually all plants have a symbiotic relationship with endophytic microbes. This relationship protects the host plant from insects due to the production of bioactive endophytic compounds. And not only fungal endophyte, microbe also makes the host plant more resistant to environmental stresses, such as dryness and cold, by production of the endophyte. So we focus the bioprotection and by conversion production. Next, host plant, Artemisia vulgaris. Artemisia is an annual plant that is grown widely across Europe and North America and Northern Africa and Asia. Worldwide, many countries we can see the Artemisia plant. That is called in English wormwood and mugwort, in Indonesia, barukina, and Japanese yomogi we call. And there are clinical herb and medicinal plant we use. Okay. In the Artemisia, Artemisia vulgaris, here, Artemisia vulgaris have a chemical structure components, bulgarin and caryophyllene oxide and artemisinin. There are terpenoids. And this is other compound. And artemisinin have anti-malarial activity. We focus this activity and select Artemisia vulgaris this time. And collection of Artemisia plant. This is parking area for bicycles and for students near the laboratory building. In here, green is Artemisia plant. This is open the blooming the flowers. The flower time we collected the Artemisia plant because they are thicker than the other time. So we collected this time Artemisia plant. As I explained in previous slide, in Japan, Artemisia plant in the ground level, but in Indonesia, grow in mountainside over 1,000 meters above sea levels, top of mountain. That is quite different of environment, but the leaf and the pollen of flower, they are similar. We think this plant is same by morphological. And by DNA sequencing, also both nucleotide sequences were same. So we can decide Japan and Indonesia plant of Artemisia is same plant. And then identified by ITS region and Matoke region. It is closely related Artemisia indica and vulgaris. Matoke region also closely Artemisia vulgaris, and Matoke region have 9 bases in Artemisia indica. So we decide this plant is Artemisia vulgaris. Okay. Consideration of haplotype study of Artemisia species, TRNH to PBSA region. There are insertion of 16 bases. So we decide in southern area of Japan, island area. It is evidence of Japanese Artemisia is southern part of Japan. Okay. A result of identification of plant, this morphology is same and nucleotide also same. After that, identified Artemisia vulgaris. Okay, next, I try endophytic isolation from Japan and Indonesia. Indonesia part is doing by Professor Andria. Artemisia vulgaris and tissue is collected and surface distilled. After distilled surface tissue is on GM medium, including chloramphenical. After growing colonies isolated 2017, 25 strains, 2018, 18 strains, 2019, 13 strains. In Indonesia also 2018, 9 strains. We could obtain many endophytic fungus. After that, identification by ITS sequences. Totally 18 genera of endophytics. 3 genera, Coledotricum and Alcinium and Giaporte. 3 genera is common of a different environment. These 3 genera is specific of Artemisia vulgaris we think, and others are infected from environment. Okay, next, screening of bioconversion ability for artemisinin. Isolated endophytic fungus, inoculated, and rotary shaking cultivation. 14 days after we add artemisinin 100 milligram and continued cultivation again 14 days. After that, homogeneated biomasses and extraction ethyl acetate and evaporate TLC. Disappeared the artemisinin spot. There is identified to bioconversion. Next, modified cultivation and extraction. Bioconversion ability fungus also inoculated cultivation 14 days. At first 0.1 milligram, and after 2 days, more many artemisinin added, so that we can reduce the incubation time 14 to 7 days. Next, bioconversion product by Coledotricum species. There are these peroxide bridge is converted to ethyl bridge and hydroxylation. But pneumonia and homopsis keep the peroxide bridge and hydroxylation. In major endophyte and before slide, in major endophyte do the change peroxide bridge to ether bridge. Minor endophyte is keep peroxide bridge. And after keep peroxide bridge is high antimalarial activity, higher than artemisinin 6 and 7 compound. There are different reaction pathway. In summary, in Japan and in Indonesia also, same plant, Artemisia vulgaris collection, and these fungus is isolated, mainly Cryptotrichum. Major endophyte by converted peroxide to ether bond, and minor endophyte it keep peroxide bridge. There are different reaction pathway. Okay, next, bioproduction of endophytic microbes. Same procedure, we can isolate certain kinds of endophytic. There are also cultivation in PDB and extracted ethyl acetate and SARS-CoV-2 assay. In the SARS-CoV-2 assay, we can find 1, 2, 3, 1, 2, 3, 4, 5. 5 extract is positive by cytopathology test and antiviral level quantitation test. That is B2 and E2 and H1 and L3 and O1. And SARS-CoV-2 3CL protease inhibition assay about that candidate of extract of endophytic E2 and L3 is positive curve of, and IC50 is 244 microgram per milliliter and 95 microgram per milliliter. There are all minor endophyte, we said then. They are not so good, but in the bioconversion, but this time useful against SARS-CoV-2 activity. Next, we try metabarcoding analysis in Artemisia vulgaris. At first, collected plant tissues, leaves, stems, roots, and collected soil. There are freeze-dry and DNA extraction. Extracted DNA is 2 times PCR, and PCR product is metabarcoding analysis using NGS. Result of metabarcoding analysis of endophytic fungus abundance about classes. Each tissue of Artemisia vulgaris in the leaf, stem, root, almost same composition, but cultivar soil is quite differently. Next, alpha and beta biodiversity comparison of fungi existing in each Artemisia vulgaris tissues. Most higher diversity is stem inner. Next is root. And the root beta diversity is far from stem, whole, skin, inner, and leaf. And then abundance of 6 fungus genera in each Artemisia vulgaris species in the Aspergillus and Cladosporium, Coretotricum, Cryptococcus, and Giaprote penicillium. In the Coretotricum, its stem skin is more high abundance, about 50%. So we want Coretotricum genus, we try to stem skin. And if we want Giaprote species, we try to root isolation. Okay, summary 2. In Artemisia vulgaris, isolated 30 kinds and white, there are screening for anti-SARS-CoV-2 activity and positive 5 kinds measuring 3CL protease for SARS-CoV-2, E2 and L3 is positive. And metabarcoding analysis of Artemisia vulgaris, stem, leaf, root, all same composition, almost same, and cultivar soil quite different. And Coretotricum species are highest abundance in this is stem of skin. So if I want Coretotricum species, do the isolate from stem skin. Okay, in the future, investigation of new artemisinin converts, artemisinin 2, more new converts, and identification of anti-SARS-CoV-2 components, culture extract to chemical structure, and application of endophytes to plant ecology for agriculture and some of health check of plant. Thank you very much. Thank you for attention.
Thank you, Maehara Sensei, for your insightful presentation. So we'll be having a discussion session for the next 7 to 10 minutes. So I will be giving a chance for 2 participants to ask questions to Maehara Sensei. I think also for those who join from Zoom or from Zoom especially, can ask question using a chat box available there. So the questions, any other questions from the participants? Yes, Prof. Partom, microphone please, Prof. Partom.
Thank you, the chairman, for my times. Okay, Dr. Maehara, Maehara-san. Okay, you saw the one compound fulgarin. Yeah, you isolate and identified from Artemia vulgaris. If we isolate from Artemia annua, because we know Artemia annua so very famous long time ago for anti-malaria. Is you can isolate the fulgarin from Artemia annua?
Yes, already.
Oh, already?
Yes.
Also. Oh, okay. Okay, second question is, you have some endophyte microbe from Artemia vulgaris from Indonesia and from Japan. How about the climate, the plant? Okay, yeah. And then if we isolate my endophyte microbe from Artemia annua, we can same product buy from Artemia annua. Thank you.
Yeah, that is not same, I think. Yes, at first, different species have different endophyte. So product is also different, I think.
Okay, thank you.
Okay.
Enough, Prof. Partom?
Okay, enough.
Okay, thank you so much. So next question. Yes, Bu Ruli.
Thank you very much for very interesting presentations. This is not my expertise, but I'm very interesting into your research result. It was said that the alpha diversity for the endophytic bacteria and endophytes is the higher in the inner skin of the plants. So I was wondering, is there any specific reason for that? That is the first question. And then the second one, I actually assume that the endophytes, the endophytes has a higher diversity in the roots because it was close to the soil rather than in the stem that was farther than compared to the roots. So is there any specific reason for that? Thank you.
Yes, that is a very interesting question. Coretotricum species is highest abundance in skin. That is some of meaning. But now I cannot understand that mean. So now continue this research together with Professor Andria. So, and to find some of reason I want. Thank you. And Coretotricum have many production shows that SARS-CoV activity also. So they are can combat to environmental stress and for example bacteria and virus and fungus. So they live in skin, I think. Thank you.
Thank you so much, Maehara Sensei. Bu Ruli, it's okay? Thank you so much. So, last opportunity. Any other questions from the audience? Okay, please, Bu. Please introduce yourself also.
Thank you, moderator, for the time. Good afternoon, Sensei. I would like to ask about your interesting presentation about the Coletotrichum, actually. And we know that based on your result in the metagenomic analysis, you have a Coletotrichum in your all sample, actually, right? Even in the root sample, in the near surrounding soil as well. So my question is this Coletotrichum in the endophyte in the plant is coming, the source coming also from the soil, or it's a different Coletotrichum since it's endophyte? And the second question, we know that the Coletotrichum itself, you can isolate and then you can develop in not endophyte, not like outside the plant. And how about the compound, the active compound of the medicinal compound? Is it still active? Is it good enough if they are developed alone, or they need to host plant to make a bigger active compound for the disease? Thank you so much.
Thank you. Question. First question is soil and other tissues endophytic is quite a different reason. That is of course, soil from plant infected from soil, but not only soil. I think this is my idea, but not evidence. So math, yeah. So I think plant can select the microbes for benefit plant service. They can select the microbes. So quite different composition today. And second question, surface endophytic is called epiphyte. There are also produced some of production, but in my study is focusing endophytic because endophytic have an interaction together with plant, so plant ability obtained to microbes. So we focus the endophytic. So I don't know about the epiphyte, sorry.
Okay, Sensei, thank you so much. I think this is the last how say questions from the audience to you.
Okay.
So ladies and gentlemen, I think we could stop the session, today's sessions, current sessions, yeah. And then before that, I will deliver some information that has been delivered by Maehara Sensei. So one of the most important thing is Maehara Sensei research have revealed the anti-SARS-CoV-2 activity from the endophytes isolated from the Artemisia plants. Yeah. And also, we do hope that the future research planned by Dr. Maehara could get success to find the active substance possessing anti-virus activity, especially for the SARS-CoV-2 activity. That's all from me. So before that, again, please give a round of applause to Dr. Maehara for the nice presentations. Thank you, Sensei.
Thank you very
Arigatou
much.
gozaimasu. And also, please, I pass the mic to MC. Thank you so much.
Give applause to our speaker and our moderator in this session. Wow. Okay. Distinguished participant present here and online participant. Let's move now. I would like to invite Dr. Yayan Wahyu Chandra Kusuma from the Research Center for Ecology and Ethnobiology to lead the next discussion. Please welcome Dr. Yayan. Give applause, Dr. Yayan.
Thank you for your kind introduction. Good afternoon, everybody. I'm Koba from Kyoto University and Center for Ecological Research, Kyoto University. And it's my great pleasure to be here at BRIN and to see you in face-to-face, finally. And I appreciate all of the committee members who organized this wonderful workshop today. So I'm working on a stable isotopes, and 2 years ago, yeah, we welcome two scientists, Kusuma-san and Shida-san, to assist with their stable isotope use. And it's my great pleasure to be here to see how they use a stable isotope at BRIN. So today, I will take the full advantage of being a second person to talk about the stable isotope after Dr. Tayasu. So I will skip most of the slides and I try to focus on the basic fundamental information I would like to deliver to you for your future research with stable isotope and the future our collaborations. So I try not to present my own data, but I will try to introduce some of test sensitive research instead. So some of you are familiar with a stable isotope, but well, my first message is stable isotope is just stable, so not toxic, and don't worry about it. This is the cartoon. This is a cartoon illustrated by Dr. Wada, who is my and Dr. Tayasu supervisor in Kyoto University. And this cartoon illustrates that a 50-kilogram person will have about 225 grams of stable isotopes like C13, O18, O17, deuterium, blah, 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 blah. So everybody, including me, you, everybody will contain the stable isotopes, and most of the materials in the solar system also have them. So it's common, it's not toxic, it's very easy to use. So don't worry. Whenever we talk about isotopes, we consider the radioactive, but these are very stable and not radioactive. So don't worry about it. So for the conservation and the sustainable use of the biological resources, it's very important to stimulate the proper use of them. And to stimulate the proper use of the biological resources, sometimes it's very important also to identify the origins and identify the history of your target materials or target animals, target plants. So as Gono sensei mentioned, we have several approaches like morphological analysis and DNA. And as Gono sensei mentioned, the DNA is now a golden method to identify the origin, and like Maehara mentioned, phylotype would be the very, very common method in these days. But I want to focus on the use of stable isotope to be a kind of complementary method to fill in the gap for the information about the origin and the history of our target materials. So, well, I've heard that probably at BRIN, nitrogen and carbon isotope technique would be started. The use of N15 and C13 will start. So I will try to focus on these isotopes. But as you may know that in Tayasense's talk, we have so many isotopes to be used. So if you are interested in like strontium or oxygen and the sulfur, please ask Tayasu about it. So I just try to focus on the carbon and nitrogen. And since the concentration itself of the heavy isotopes like carbon 13 and N15 is quite low. For example, in nitrogen, in the case of nitrogen, the heavy nitrogen isotope N15 has about 0.37% for you and for me and for everybody. So very low. So it's not easy to illustrate the high and high or low signature. So we instead of the use of the normal percentage notation, we use this kind of weird delta notation. So for nitrogen isotopes, we call it delta N15, and for carbon, we call it delta C13. But this is my second message. This is just a proxy of the heavy isotopes. For example, if we mentioned that this has a high delta N15 value, that means your target compound would have relatively higher N15 concentration compared with the others. Okay, so it's just a concentration of rare heavy isotopes. The third message, which is very, very important before using the stable isotope, is that we have typically two approaches to use the stable isotopes. First one is that we call the labeled isotope compounds to use in your experiment. This is the common way to use the stable isotopes. But the method that Tayasu and I are working on is just the second one, exploitation for the natural variation of the stable isotope concentration. This is rare. So in most of the cases, when you talk about the use of stable isotope, this is what you have to, this is what you have in your mind. Now, this is the example of the very famous experiment to identify where oxygen comes from via photosynthesis, from water or from CO2. It's really, really hard to define. It's really hard to identify which processes are occurring in the photosynthesis. Because oxygen is just oxygen, CO2 is just CO2, and water is just water. In this experiment, they labeled water or CO2 with this kind of labeled oxygen O18. Then they found that from this experiment, they measured the high concentration of O18 in the oxygen produced by the photosynthesis. On the other hand, in this experiment, no oxygen O18 in the O2. So this is very, very clear and very, very straightforward to interpret what kind of processes are going on in your experimental setting. Okay, but this is just an experiment that they use the very high concentration of the rare stable isotopes. Okay. So you can purchase so many compounds labeled with O18 or labeled with N15 or whatever. But the question is, how to use them. This is a graph showing the delta O18 value as an x-axis. The y-axis is the O18 concentration and the O16 concentration. These three are kind of identical. But what I would like to see is this part, The delta notation is stretching the very, very tiny range in O18 or N15 concentration variation. So roughly speaking, this unit and this percentage unit is totally different. If we convert the delta O18 unit expressed in per mil into the normal concentration notation in percent, 0.1 per mil equals 0.002 to the very, very tiny difference we want to stretch to express the variation in delta N15 or delta N15 and O18. So simply speaking, we cannot carry out the two kinds of experiment in the same single room. So this is another cartoon showing that the C13 concentration in percentage and the C13 concentration in delta notation. So whatever you sample from your ecosystem, the range of your sample would be like here, in the solar system from -50 to +50. That's the maximum. But if you convert this data into the normal percentage, that range is very, very narrow, right? So if you want to use the labeled compound like C13 labeled glucose in your laboratory, tiny contamination of your sample to measure, if you want to measure the natural abundance of C13 for your target plant. And if you contaminate very, very tiny amount of this labeled compound, you will get a very, very unique and very, very interesting data, right? So in this case, when you analyze a 0.5 milligram like this, of your plant, and if you had, I'm sorry, you cannot identify this contamination, but if you had 0.0001 milligram, very, very tiny amount of labeled material contamination, you will have the very big change in delta C13, like from normal honey to weird honey. So my note is, please do not carry out these two types of experiment in the same room or same balance or same machine. That can induce the analytical error, very serious analytical error. And if you want to do some forensic studies, and if you want to identify this material come from here and there, it's critical. So please do not try to measure these different types of isotope samples in the same way. Okay. Oh, so the reason, I mentioned that the concentration of N15 is roughly 0.737% for all of us. But if we look into the detail in the concentration, everybody had slightly different concentration of N15. The reason why you and I and lots of things can have the different isotopic ratio is the reason, the consequence of this isotope effect or isotope fractionation. Let's think about some reaction from A to B. Okay, so the reactant will be converted into the product B. During the course of this reaction, for the nitrogen, we have N14 and N15 isotopes, and N14 is slightly lighter, and that can react faster, slightly faster than N15. So at the middle of the reaction, we can find the accumulation of lighter isotopes that react faster than the heavy one. So in the reactant, the heavy isotope will be accumulated or preserved or left. So at the end of the reaction, the ratio of N15 and N14 of the reactant A is preserved in the product B, of course. But at the middle of the process, we can see the difference between, we can see the difference in the accumulation of N14 or N15 for product and reactant. That is a rule of thumb. The faster, the lighter isotope is, the faster they react. It's very simple. Okay, that can happen everywhere in your stomach, in the environment, in the solar system. That's why many compounds will have their own unique isotopic signature, depending on this kind of processes. Okay. So, as Tayasu mentioned, one of the most important, easy to understand example is the delta C13 difference between C3 plants and C4 plants. It's depending on the photosynthetic pathways for C3 plants and C4 plants. And as Tayasu and as in the discussion, in the case of the honey, in most of the cases, we find the delta C13 of honey like here, depending based on the C3 plant. But some bad guys try to add the sugar produced from sugarcane C4 plant. This data should be sifted like this. So such kind of information can be used to identify the origin or I identify the origin of honey or identify the contamination of something. Okay. So basic idea of the use of stable isotope for our analysis, for our approach, we call it natural abundance approach is like this. This is another example of isoscape measured and simulated in the United States. As Tayasu briefly mentioned, the hydrogen isotope ratio of precipitation or groundwater, river water, depending on the temperature and the precipitation amount of latitude. So we can draw this kind of isoscape by direct measurement or simulation. And once we got this kind of isoscapes, and once we decide to study what kind of animals or what kind of organism you want to study, like in this case, if we want to track the migration route of some target animals, we can take the advantage of these isoscapes. And once we confirm that which body parts can preserve the isotopic signature, in this bird's case, they confirmed that the hydrogen isotopic ratio of available water can be preserved in the weather. So they measure the hydrogen isotopic ratio of weather and see where they went and where they went back. In this weather, the history of migration is preserved. So such kind of approach can be done at BRIN and anywhere, as long as we can identify this kind of beautiful isoscapes. Okay. So, I know you cannot see this, but we can apply this method in many ways, like antique and art materials and, food materials, including honey, and I cannot see, but some drug materials, marijuana and cocaine, and yes, of course, wildlife and some chemical. So please take a look at this table from the slide I shared, or just you can check the leaf. Oh, I cannot see you can see this kind of book. We have so many references regarding this kind of thing. Okay, so stable isotope approach is kind of common and very easy to use as long as we can identify the isoscapes. So let's go back to some specific examples. 2 years ago, Kusuna-san mentioned she will work on the agarwood, and Shinta-san, I cannot see that probably she was working on the parrot. Okay, so I try to dig out the previous studies regarding these topics, which I'm not familiar with. Anyway, this is the only data I got for parrot. This is the carbon isotopic data and nitrogen isotopic data of birds collected in one single forest. So amazingly, these 22 bird species have kind of similar C13 and N15 signature, indicating that they are sharing the same kind of diet in this forest. And happily, they found very distinguished pattern for these 1, 2, 3, 4, 5 species. That is good start. And especially, this is their target, cave parrots. So at least from this data, they can mention that probably they have kind of unique diet compared with other birds. It's good. But the question is, what's more, can we dig out a little bit more from this data? So going back to the Tayasu sensei's, the talk, regarding carbon-13, the diet and the host or prey and predator have the almost same carbon isotopic ratio. It's preserved. But for nitrogen, due to the excretion of nitrogen, that can induce the loss of N14. we will accumulate slightly the N15 by 3.4 per mil by the delta notation unit. So, like one trophic level induce the changes in about 1 per mil for C13 and 3 per mil for N14. So that implies if your target animal has a high trophic level, your target material will have the high delta N15. Okay, such kind of information, including the difference between C3 and C14 plants. Okay, oh, thank you. So we can speculate the diet of these birds. Okay, so for example, these birds have very high delta C13, implying they rely on C3 plant more than other species. And for this species with high delta N15, probably this guy would rely on insects or fish with slightly higher trophic level. And in the same way, in this paper, they discuss the diet of this Cape parrot as a fruit that would be expected to have low delta N15. Okay, so that kind of interpretation can be possible, but as you might feel right now, these interpretations cannot be derived directly from these iSpeak data themselves. We need more supportive information about the background of the iSpeak ratio and about the feeding yield provided by the direct observation. So stable isotope technique is not perfect at all. We definitely need more information to help interpretation of this data. Okay. So this is one of the examples by Tayasu did, several years ago, about 10 years ago. This is an example of the diet identification of bear in Hokkaido Island. This paper includes several advantages of the use of stable isotopes. First, they used a specimen collected long time ago. It's very nice to reconstruct the diet from specimen collected long time ago. Okay. Second thing is it's kind of easy to measure the stable isotopes. The third thing is, they can discuss the sample collected from northern part of Hokkaido Island, the western part of the Hokkaido Island, the southern, eastern part of Hokkaido Island, and collected from different time period. So once we draw this picture, we can, when you measure the specimen and plot your data on this N and C map, you can identify, when your specimen will be collected, or in more detail, with the help of these play data, you can calculate how much the old bear relied on corn or relied on a salmon, such kind of diet information we can derive. So, stable isotope approach is not perfect, but this kind of information is so unique to get. So, it's very nice if you can use this technique complementary to other methods like DNA and morphological analysis. So time running. So let's go back to the crops or plants. This is one of the big, nice example for crops. Bell pepper. They measure the delta N15 and delta C13 of bell pepper, but are purchased from Spain, Netherlands, and Germany. And you can see a very nice, clear pattern among three countries. I don't know why, but this is very, clear. And once you get a bell pepper and measure your data, then plot in this CN plot, you can identify if your bell pepper is like Spanish or Germanish or whatever. Such kind of thing can be happening. But we want to dig out a little bit more. But the question is how to interpret this data. Oh, I don't know if you can see it. Even in the same place and the same species, but a different agricultural application can make that delta N15 different. This is the case for the tomato and lettuce and whatever with conventional inorganic fertilization agriculture and organic agriculture. So this is very clear case for tomato. So tomato from organic farm have very high delta N15. So this can also provide us some information about what kind of application agriculturally the farmer did. This is very important. This will be very unique information. But even in the organic farming, in this case, they use a different manure. And different manure induce the different iceberg signature. So stories are getting more and more complicated. Okay. So I found only one single data for agarwood. Agarwood would be the raisin, right? But this data is not raisin, but leaf data. So this is the agarwood species, delta N15 and delta C13. And they try to interpret these variations along with the foliar nitrogen concentration. Unfortunately, these values are very, very normal, very, very common, not unique data compared with the global data set, right? This is the range for this value, but this is a collection of the plant data all over the world, more than 10,000 data. So very, very unique. So I cannot tell, if this data is so unique or not to be able to identify the this is agarwood or this is not agarwood, such kind of thing. It's important. It's impossible. And if we want to dig out more detail for plant delta N15, we should concentrate lots of things. This is a review written in 1996, almost 20 years ago, but the situation is still the same. We have to take care of these sort of parameters to interpret the delta N15 of plant. For example, if the plant have shallow root depths, they will enjoy the nitrogen at the surface with low delta N15 compared with the high delta N15 nitrogen at the deep soil. This is caused by the nitrogen decomposition. During the course of the decomposition, lighter nitrogen would be gone as gas or water. So along with the decreasing the concentration, the delta N15 is increasing. So that's why plants who enjoy the nitrogen at the surface, they will have lower delta N15. While some plants try to have the root at the deeper soil, they will have the higher delta N15. So this is one of the examples. This is the delta N15 of plant, and this is the plant maximum root depth. So lower plant has a shallower roots, but higher delta N15 plant has a very high rooting depth. Okay, such kind of thing we have to think about, consider when we have to interpret the delta N15. So same goes for the mycorrhizal, non-mycorrhizal, and what kind of nitrogen the plant will rely on. So it's so complicated. So my final message is, it's nice to start to obtain such kind of data, especially for the endangered species. It's very, very important to grasp. It's very important to get insight into the diet. But the diet analysis based on the stable isotope is completely bad, or just incomplete. And we need more information about them. For example, if we want to talk about the stable isotope analysis, we definitely need the stable isotope signature of prey, or stable isotope of signature of nitrogen sources for plants. Okay, so we need another additional data to interpret, to dig out more information from these stable isotope signature. But at the first point, it's really nice to draw this kind of picture to see if your target organism, you will have some unique characteristics compared with other organisms. That is a good start, and that is what we can do kind of easily. So Tayasu Sensei and I will be here until Friday. So please catch us whenever you have some question about the use of stable isotope. That's all. Thank you for your attention.
Thank you, Professor Koba. Would you please to join me on the stage, because time is running up. So I would like to combine the talk. So we will have discussions at the end of the talk. So please hold your questions later on. I see some participants already writing some things. Maybe a lot of questions will coming up. I would like to also introduce you Dr. Ruliyana. He is a researcher for ecology and ethnobiology, BRIN, Indonesia. She obtained her undergraduate degree and also master from ITB and continued a PhD in Hiroshima University. We had a lot of involvements in international projects, and also as an Indonesian delegate in many international works. And she now will continue with the stable isotope, working with agarwood, talking about exploring stable isotope analysis for agarwood research, challenge and opportunity. So it's still in line with the previous talk. So please, Dr. Ruliyana, the floor is yours.
Thank you very much, Dr. Yayang, for the nice introductions. Good afternoon, ladies and gentlemen, participants. Actually, honestly, I'm a bit nervous to standing here because stable isotope is a new playground for us. We still have a lot of things to learn, and we would like to learn from Tayasu Sensei and also from Koba Sensei. So today, for this talk, I will focus more to the exploring stable isotope analysis for agarwood research in Indonesia, its challenges and the opportunity that we can seek with this research. Before we go to the stable isotope analysis, first I would like to introduce you all to what is agarwood species. So agarwood is actually a non-timber forest product export commodity with various trade names. In Japan, it was known as ginkgo. In Indonesia, it was known as gaharu, and in Arabic, it's oud. There are several others named. And in Indonesia, actually, there are 13 species distributed along Indonesian islands with two major genera, which is Acularia and Gyrinopsis. And in Indonesia, the distributions of the species is very distinguished. So there are species that was distributed only in the western part of Indonesia. There are about 4 species, and 9 other species was distributed only in the eastern part of Indonesia in several islands. So in this picture, we can see that example for Aquilaria malacensis that was distributed on the western part of Indonesia. This is the natural distributed that I generate from the JASTIP data set. But for the plantations of this species, it was already planted across Indonesia, even up to the eastern part of Indonesia. And the same thing goes for Gyrinopsis verstigi. So this species only distributed in the eastern part of Indonesia. So it's in the Lesser Sunda Islands, the small islands over here. And actually there are data, natural distributions, new data from Sulawesi or Celebes, and the natural distributions also in Papua. But again, the plantations of this species was found in Maluku, and then also in Sumatra and Kalimantan. So for the plantations of this gaharu producing species producing gaharu is distributed along Indonesia's islands. That is the things that we have to resolve for Indonesia, and why we choose the gaharu, and we need to make the non-detrimental finding for gaharu is because the trade of gaharu is very one of the species that give economic value for Indonesia in higher rate. So if we see here in this left graph, this is the national revenue acquired from the trade of flora and fauna. So it's included gaharu also. The amount of the money that was played here is very big. That is why the government is very supporting in developing methodology for identifications or for forensic research related with the wildlife species. And this is the harvesting of agarwood that was put on the trade market. The trade of the gaharu or agarwood is conducted with limitation quota. So there are quota decided each year how many kilogram or how many ton that can be exported outside Indonesia. And the value of the quota and realizations of the quota for export of Aqualaria macalacensis is decreasing along the year. This is the data for the wild or the from the nature, from the forest. Because one of the reason is because the scarcity of the species of the individuals, and also because there are many also produce products in the market that come from the plantations. So the plantations of agarwood the plantations of the plant producing agarwood is already successful in some part of Indonesia, and they can be exported. And for Gyrinopsis verstigi, the realizations and the quota is relatively stagnant, but in 2021, the value is very increasing, very distinctive here because of the production from the decaying log. So there are another type of product for gaharu, for agarwood. Next, this graph shows why we have to make actions to ensure the sustainability of wildlife species in the trade market. Because based on the review conducted by IUCN, the research shows that more than 900 species at risk of animal and plant not covered in global trade protection. So for the endangered species trade, it's all regulated under international bodies called as CITES. And the number of species threatened is increased 4 times in the local and national market rather than international market. So this is what I said previously. The Convention on International Trade in Endangered Species of Wild Flora and Fauna makes sure that all trade of wildlife species or flora and fauna can be traced. So traceability is very important to know the tracking mechanism and then the change of custody. And for this, identification is very important. The second one is the legality to ensure the timber or log legality, and sustainability to ensure timber or product come from sustainable source. Next, this is the challenges that we identified for the agarwood in Indonesia in case of identification of agarwood in the market. So most of the product of the agarwood
is not in the form of log. So it is very difficult to identify the species only by anatomy or morphology. Most of the product in the market is in the form of chips, 52%, or powder, 41%. So only 1% product traded in the form of log. So first problem is the origin of specimen collected is questionable. We have a lack of clarity regarding the geographical origins, whether is it country, because as we see in the previous map that Aqualaria malacensis also distributed not only in Indonesia, but other Southeast Asia country, or the islands in Indonesia, because we have to control from which island the species was, the product was harvested. And then the second one is difficulty in distinguish whether the product is come from the wild, it means from the natural distributions, or from the plantations. Because in case of plantation, there is no limitations of quota. So so we can export in a large amount of volume compared to this the wild product. Wild agar would come from the wild. And then the third one is the species identifications from byproduct, because most of the product is in the form of chips and then powder. Is it a bit difficult to identify its species? Sometimes it was mixed, and this is also challenges. Why this is important? Because it's related with the legality of the product. Whether because the product was listed as a species, so we have to sure which species was imported was exported. And this is compilations of several methodology that have been conducted to develop for identifications of wood. It's not only for agar wood, but wood as in the general term. There are several methodology from the conventional one, whether it is morphology based on macroscopic characteristic, and then anatomy. And then there is a methodology for machine vision or machine learning. We also developed this. And then there is methodology for mass spectrometry and near infrared spectroscopy, stable isotope analysis, radiocarbon, and DNA barcoding, population genetic, and fingerprint. All the methodology has different characteristics. Some is commonly used and very easy to use, but others is very difficult. For agarwood, there is data from Espinosa that using mass spectrophotometry to distinguish whether Aquilaria timber is from plantations or grow naturally. And then another one using DNA barcoding that we also use in BRIN for species authentication of agarwood, which is Aquilaria products, but only until the species level. The questions is come for stable isotope that has possibility to identify the geographical origins of this of the product. The mass spectrophotometry and the NIRS and also population genetics can also be used for to identify geographical origin. But for population genetic, it will be difficult if the samples that we use is a product from the market. The near infrared spectrophotometry and then mass spectrophotometry and stable isotope is very possible to be conducted. And I guess based on our limited experience, it's very easy actually to analyze, to conduct the research for a stable isotope. The difficult thing is to analyze the data, right, Sensei? To interpret and analyze the data. How we conduct it, maybe we can learn it very fast for stable isotope compared with the others methodology. Next, this is the state of techniques that was available in Indonesia for the stable isotope. The equipment is available, but it has not been tested for wood. Actually, more of the research related with stable isotope in Indonesia is related with food and agriculture to trace whether the organic or non-organic product from plants, and also for food to trace the origin of rice, for examples. And also the result of the analysis have not yet been verified. BRIN now conducting this wood anatomy. We already launched the wood sample collections and program for this. I forget the name. Do you remember, Payayan? I forget the name. This is from Forda, the Ministry of Environmental and Forestry, but already moved to BRIN, so it's now under BRIN. And then the NIRS is now undergoing, conducted in IPB and also in BRIN, in the Research Center for Biomass. And then we try also to conducting for stable isotope analysis. Next, this is I think I can skip this one because we already get a lot from Tayasu Sensei and Koba Sensei. But for me, the basic simple principle for stable isotope is you are what you eat. So that the isotopic composition of an organism tissue reflect the reflect the isotopic composition of its diet, so what the plant intake and or environment. And next, this is the ongoing study that we conducted. We still focus on the collecting samples and explore the wild species, the wild samples, because it's very difficult to get the wild samples, the fresh sample from the wild, and also the plantations. So we also identified which area already has a plantations and whether the plantations is already productive or not. So we already collected several samples from Sumatra, Jawa, Kalimantan, and Maluku Islands. And we also collecting environmental data, the water and soils. Water only in several areas, but soil is in many areas. And collections also for various products in the market. And we conducting some capacity development and some trial and error, and many of the error come to our trial. And then this is the simple methodology that we use. We still only have one standard, Sensei, and I try to apply for another standard, then we're still waiting for the standard to come. And this is the number of samples that we already collected. There are many samples, so it's ready to use for analysis actually. And then we conducting capacity building, as Koba Sensei already mentions. There are two young researchers sent to Japan for workshop for stable isotope ecology in Center for Ecological Research in Saga under Kyoto University in Shiga, Japan in 2022. They come with a lot of things and then a lot of questions for our research. What should we do for the next step? And then this is some problems that we try to identify. So first, there are product in the market is in the form of cheap, powder, and low. It's fairly limited in log, so difficult to identify the exact species or origin with morphology and anatomy. We can identify the species with the DNA barcoding, but then only until the species level. We have a lack of deep understanding in the concept of stable isotope, especially for this wildlife tracking and origin, and difficulties to determine the best standard and in-house standard availability. We still lack in technical skill to operate the machine, the RMS, And in order to use stable isotope for agarwood tracking, database of agarwood isotopration need to be developed. So Sensei, to give you a figure out, participants, ladies and gentlemen, this is example of fresh stem of agarwood species. If I'm not wrong, this is the Aquilaria species, and this is the agarwood. This is the resin. So, however, in the market, sometimes it's not only this part was sell, but whole part of the plant, of the wood. So this is also example. So the black one is the resin or the agarwood, the one that has a very high value in the market. And for the future research, we still plan to conducting capacity building. So tomorrow we will have a discussion. There are several schemes that can be used for the capacity building. And we are planning also in developing standard protocol for stable isotope to track timber and wood, especially for agarwood and others animals, for birds, and then for the material that Prof. Gono mentioned in the morning. So the important thing is the protocol to conducting the research in the IRMS laboratorium. We haven't made this kind of protocols, and it was very important to have it. And the establishment of Indonesian wildlife database for stable isotope character, and then strengthen collaboration with neighboring countries that share same species. It's also very important so in order to get samples from different geographical origins. And then this is the opportunity that we have in Indonesia. There are a lot of machines actually. I'm a bit surprised in Indonesia. This is very expensive machines. This is the machines that is available in BRIN, in the genomic laboratorium. So Sensei will, all the professors will visit this location tomorrow. There are 2 machines in BRIN, one in Cibinong and one in Jakarta. And 2 was bought for the National Narcotic Board, the BNN, the new one. If I'm not wrong, there are one in ITB, the one that was come with the result that was mentioned by Tayasu Sensei. And also, I heard that the Ministry of Health is planning to buy these machines. So Indonesia have a lot of machines, but we still lack in the capability, and we need more capacity building on this part. And a lesson learned for me and for our team, JASTIP is an example of collaboration that allows its member to engage with other experts and researchers in their field. So even though we've overcome the COVID pandemic, we still can make some communications with via the online with Zoom. So this is the first time for me to meet Tayasu Sensei and Koba Sensei, even though we already discussed for 2 years. But then again, the online discussion has a lot of limitation compared then if we can meet directly. So we have to meet many times, Sensei. And then this kind of collaborations also open opportunity for broader networking. There is no limitation. We can like jump and ask other partner to conduct one part of the whole project, and then we can collaborate. For example, for the samples that we get from Maluku Islands, we get the sample with collaborations with University of Patimura, and then we also apply for JASTIPnet with University of Patimura, and also together with the local forestry office or BKSDA. And this project also give opportunity for improving capacity with training, seminar, and workshop. This kind of workshop, and also workshop that these 2 beautiful ladies come in to Japan. And this is the end of my presentation. Thank you very much, ladies and gentlemen, for listening of me. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Ruli. Dr. Koba and Dr. Ruli, please join me on the stage. So it's time now for questions and comments. we have about 6 minutes left. So I would like to take for the first 2 questions or comments from the audience in this seminar hall. Please raise your hand, mention your name and also your affiliations, then ask your question or comments, please. It's a quick, okay. We have one question from Professor Tayasu first, okay.
I'm Ichiro Tayasu from the IN, and I'd like to ask Ruliyana-san about samples. And you mentioned that you have sampled from 4 islands. 4 islands means 4 islands with 1 plantation and some other wild populations. So what kind of sampling systematics?
Yeah, please directly.
Thank you for the question, Sensei. It's depend. Or sometimes we can get a sample from the wild individuals. The problem is it's very difficult to get the wild individuals because the trader didn't want to give us samples because small amount of samples is it's have a huge value of money, so very expensive unless we pay for the sample, so to get several samples. But for several area from Sumatra and then for Maluku, we get the fresh samples, the natural, and then the plantations one. So it's depend on the locations. There is more detail on the data on which species already identified up to species level, which samples that not yet identified up to species level.
Okay, thank you.
Thank you, Professor Tayasu and Dr. Ruli. Is there any other questions from the audience, or maybe from the Zoom participants? Are there any participants? Please raise also your hands if you have any questions from the Zoom participants. If you don't have any, oh, okay, there is one. Oh, Mr. Bramantio, please unmute yourself and start ask question.
Can you hear me, Mr. Yayan?
Yes, please start here, Mr.
Yeah,
Bramantio.
thank you so much for the presentations, Koba Sensei and Tayasu Sensei. I would like to ask that I remember Tayasu Sensei has ever published a paper in termites with the analysis of the stable isotope as well with Takematsu Sensei. If I'm not wrong, they conduct a analysis on termite from Thailand at that time, back then. So at that time, Tayasu Sensei analyzed about the different feeding habits. So there are some termites eating from soil, from wood, and then from other food sources. What I would like to ask is that, can we trace if the termites come from forest area or urban area? Forest area means that they have a lot of humus and trees surrounding them. And then the one with termites come from the urban area, which only consists of structural wood or different surroundings, such with the compared to the forest one. Can we analyze the stable isotopes, and then can we just like understand how they move or migrate, and then how long they have been stayed in urban or forest area? Because this is related to the efficacy of termites in Indonesia. So in Indonesia, if we want to produce a biopesticide or pesticide or chemical, we need to test it with the termites. However, right now, the species that is used for the test is related to the forest only. And the species which is related to the urban one has not been used until now. So that's why I am asking if we can understand the migrations, or not the migrations actually, but how long they have been stayed in the urban or in the forest area by looking at the stable isotope. You got my questions, Mr. Yayan.
Yeah. Yes, Mr. Dharmayanti, at this section.
Thank you so much.
How to differentiate between stable isotopes termites from the forest and out of leaves in the urban area, and can it be traced, and how long actually the termites that exist in the urban area they came from the forest, how long they have been in the urban area? Please, Koba, thank you.
Pass it to Tayasu.
Interesting.
Nobody
Okay.
knows that he was a
Time science. Okay.
Yeah, my PhD work was on termites.
Thank you so much.
Okay, that's I suppose 2 points are important. And usually when I studied in Australia or Africa, the forest savanna transition is from C3 to C4 plants. So in that case, it's quite easy to identify which termites eats C4 forest or C3 forest or C3 grassland, C4 grassland. But in the case of only C3 forest with C3 woodland, in that case, it is difficult to distinguish between the two. Although, as I mentioned, carbon-13 value will change dependent on precipitation regime, dependent on the stomata's openness. So usually inside the forest, carbon isotope ratio is lower than the other parts, and dry area that's close to open to grassland is higher carbon-13 values. In that case, we can distinguish between the two. However, as I mentioned, carbon-13 value varies only slightly within C3 plants. So the dramatic change with C3 to C4 is easier. So that means that depends on the situation that which C3 or C4 plants were there. It's okay?
Does that answer your question, Mr. Dharmayanti? Seems like it's a good opportunity to initiate a collaboration project with Professor Tayasu regarding to this matter.
Yeah, thank you so much. It's a quite opportunity, I think, in Indonesia to trace how this termite moved in the forest area and also in urban area, I think. So this is the big problem in Indonesia because we still don't understand which termite is actually populated in urban area and populated in forest area. And it's make a great impact in the standard national procedure for the efficacy of wood-based product. So I think this is a good opportunity to collaborate with Tayasu Sensei. Thank you, Mr. Yang.
Thank you, Mr. Dharmayanti. I think we go back again to stable isotopes topics. Please, is there any other questions regarding to that? Because as you know, as already Ibu mentioned, we have 2 machines in BRIN, one's in Cibinong, so the other in Serpong, right? No, in Pasar in Jakarta. So 2 machines, it must be used and needs to be used frequently because it's a very expensive machine. So there are a lot of opportunities, not only using the machines that are available in BRIN, but also to collaborate with the professors from Japan. So if you have any other questions related to that, there are many topics or many research studies that can be used, incorporate these stable isotopes for your research. Because I remember a couple of years ago, I attended a workshop as well, Professor, using stable isotopes to study the sap flux and also evapotranspirations within the forest. So it brings me to a question. If we compare these 2 methods, like there are actually another machine for subflux, for instance, like subflux meter that can also measure real time as well, and combine to compare to the stable isotopes, which one is kind of more accurate or maybe easier to apply and cheaper? Which one? What do you think, Professor Kobe?
Wow, it depends. And one of the biggest advantage of the use of stable isotope would be totally invisible. So we don't have to touch the ecosystem itself. Just try to dig out the information from outside. You know, whenever the your target ecosystem is kind of vulnerable against the kind of the application like watering or shaking or collecting sample that can change the flux of the material. In that case, stable isotope would have a huge advantage because we don't have to worry about the alteration of the ecosystem itself. Yeah, but as you mentioned, it depends. If we want to think about the cost, we have to have a big debate.
Okay.
Yeah.
Okay. Yeah. But since we have the machines, might be the cost can be negotiable. So I think it's already time is already passed. So we would like to thank you to the two speakers in here. Thank you very much, and please give a big applause to them. I give back the floor to the MC. Thank you very much. Good afternoon, ladies and gentlemen.
Give applause again to speakers and our moderator in this session. Wow, very interesting. Okay. For the last session, I would like to once again invite Dr. Naraputra from the Research Center from Pharmaceutical Ingredient and Traditional Medicine to lead this last session. Please welcome Dr. Naraputra. Give plus Dr. Naraputra.
Thank you so much, MC. Actually, I'm
Thank you for kind introduction. Good afternoon, ladies and gentlemen. My name is Hiroshi Kamitakahara of Kyoto University. Today, I'm going to talk about the synthesis of high value-added products from terpenoids. And I must say, maybe you are very tired for whole day. And anyway, today I have to explain the work package 3 of the JASTIP. Now, there are two groups, and one is this biodiversity group, and The second one is Umemura Sensei and Watanabe Sensei's group, the biomaterials group. And now I will show you the member of the biodiversity group. These groups are divided into two subgroups. The one is bioprospecting group. The leader is Dr. Andrea Augusta, and isotope forensics groups, and Dr. Gono and Dr. Ruliyana-san. And now you can see the Maehara sensei, Tanaka sensei is here. And unfortunately, Dr. Watashi couldn't come to Indonesia, but hopefully he will come to Indonesia in the future. And you can see that Tayasu sensei, Koba sensei are here. And I think it is very good opportunity to have a good connection between the LIPI, not now that's BRIN, the former LIPI, and the Japanese researchers. And I have to mention that Umemura Sensei's institute, the former name is Research Institute of Research Center of the Research Institute. Research Institute for the now it's a different name, but Umemura Sensei's Institute has a long history for the collaboration between the LIPI and Kyoto University. So that's why there are many colleagues working in the BRIN. So we would like to continue such a good collaboration in the future. So last year we had INARI is expo was held in the Chibino. But limitation due to the limitation of COVID-19, we couldn't go to Chibino, but we had a poster of the WP3 there. And this slide shows history of our collaboration between the Dr. Andria and I and Dr. Takano of Kyoto University. We had probably started our collaboration in 2009. Looks young. And you can see that we have visited Central Kalimantan in Salibumi Kusuma concession area. We are visited twice. The first one is 2011 and the second time 2012. So we had collected many plant species there. He is Dr. Andrea and I, and there are some workers from the Salibumi Kusuma. We had visited some villages inside the concession area to hear the what is medicinal plant they used something. Then this shows a map of the Kalimantan. We visited this area. So there are many concession area. Logging concession is in yellow part that we visited here. And the pink stars show the place for the collecting samples. This shows resin of the dipterocarpaceae. The name is Damal. Damal resin is we collect this Dharma resin from some shorter species like this, and Dharma resin includes triterpenoids compound like this. There are many kinds of the terpenoids included in resin. So that means the resin, resin, probably the plant use the resin to protect the plant body from the maybe attack from the termite or the fungi attack or something. So that means very important compounds are included in the Dharma. So the in contrast, this one is a white birch. The Japanese name is the Shirakaba. This plant species include BRIN. So the BRIN is very important compound because that has anti-cancer activity or betrinic acid. Here, the common number 28 is oxidized, and that we called as a betrinic acid that has anti-HIV activity. So, and I have to say that this BRIN is a main compounds of the outer bark of the birch, and it includes about 20 to 25%. So I can say that is very large amount in the bark. So that's why I would like to start research on the topic of the BRIN. And the BRIN triterpenoids has very hydrophobic properties. So we would like to make the BRIN the hydrophilic water-soluble compound to prepare some drug carrier like this to prepare the surfactant. Inside the surfactant, maybe hydrophobic compound will be included. Or this one is this is a polysaccharide backbone, and the hydrophobic compound is included inside a micelle, and then this micelle will be used as a carrier for the medicine, hydrophobic medicine. So next, I'd like to start actual research, the synthetic research. So the key is to keep some key structures, chemical structure like this part or the hydroxy group part. We have to keep the structure. So that's why we select this double bond for the chemical linkage between the hydrophilic sugar part and the hydrophobic triterpenoid part. So that we use the strategy for azide alkyne quick reaction to connect the two parts, the hydrophilic part and the hydrophobic part, to give a kind of artificial saponin compound. So the next, I'm going to talk about the hydroazidation of the BRIN. So we could introduce the azide compound here. So this slide shows a previous study of the hydroazidation. So ferric, this one, ferric oxalate and sodium borohydrate will produce such a radical, and then after that, this radical will be converted to the azide compound. Maybe I should skip this table, but based on the literature, we had studied the reactions, but we had to increase the reagent to get this product too in the good yield, about 96%. So this slide shows the FTIR spectra of this compound 1 and compound 2. Obviously, you can see the difference. So this absorption means existence of the azide group. And also this slide shows the proton NMR spectra of the compounds 1 and 2. As you can see, these protons disappears in the case of the compound 2, and we can see that these protons at the carbon 30 appears, and this proton moves to the higher magnetic field. So after maybe I can skip this one. Anyway, we could prepare the cell biosol derivative having some alkyne group at the reducing end. So next, we could obtain the compound of the artificial saponin-like compound. So this is evidence of this chemical linkage between hydrophilic and the hydrophobic part. This proton appears. And maybe as expected, this compound will make the micelle. You can see you can see if we will dissolve the compound in the water, we can see this is a laser beam. The laser beam will make the make the line like this. This means some micelle exists in this water. So the diameter, this shows the dynamic light scattering data, and the hydrodynamic diameter is about 100 nanometer. So that means such a micelle can convey some hydrophobic agent to some, for example, we can inject such a compound into the human body. So, and also that we can say this compound will become the detergent, like the soap. Here you can see in concentration, the 7.3 millimolar of the compound D6 has a surface activity like this 30.8 millinewton per meter. That is, this value is better than sodium dodecil sulfate. This is comparative to the such a typical detergent compound. Yes. Next, let us move on to the synthesis of the betylene cellulose conjugate. Now we use water-soluble cellulose acetate, and we introduce the acyl group having this alkyne group here. So we have introduced such a hydrophobic triterpenoid compound by a azide alkyne chemistry, or we can introduce this one like the the octa-arginine residue. This residue have the ability to convey some compound into the cell. So that's why we would like to introduce such a moiety on the cellulose backbone. Yes, so that we have now going to talk about the synthetic method. We had used acid, and this is EDC and DMAP or dimethyl aminopyridine. It's a base, and we could get this compound in good yield. And also we had got such a compound with azide functionalized octa-arginine residue. Excuse me, I have to mention about this one. Zeta potential of this compound is plus 9.9 milli. So this means my cell has a cationic surface. So we have to talk about how we prepared, but as you can see, this is an NMR spectrum of compound 11, this one, and we cannot see not so much signals of this betrin residue. This means betrin residue is inside the micelle, so the movement of the betrin residue is very slow, and we cannot see not so much signals, but we can see the cellulose moiety here. So this spectral means such a micelle formed in the water. As expected, we can see some micelle formation. The dynamic light scattering measurement shows its hydrodynamic diameter is about 1 micrometer or 1.8 micrometer. So the colloid solution is observed here. So now I would like to summarize my talk. The key point of our research is to keep important functional group like a primary hydroxy group, which can be converted into to the acid later than later after preparation of the cellulose between conjugate. We can convert some functional group to other functional groups, and also we could prepare the betrin cell bios conjugate that will show some interesting property like the detergent or yes, actually formation of the micelle must act as a carrier for the hydrophobic drugs. So we could also use some cellulose derivative for the carrier for the other triterpenoid compound. But this cellulose derivative can be used for or carrier for other drugs, like not only for the triterpenoids, but also for many other kinds of the compounds can be introduced. So that's why if we would like to carry some functional compounds on the cellulose, we can easily prepare based on such a strategy. Okay, thank you for your kind attention. And this is the picture which I took last year. Our institute, the Graduate School of Agriculture, and this is Diamond Mountain. The coloring of this year is not so yellow now, but hopefully you can, if you visit Kyoto, you can enjoy the coloring autumn season now. Thank you for kind attention.
Presentations. It's very firm and very clear, the presentations about the how say the steps used by sensei to increase the hydrophilicity of the substance. So I will open maybe one terms of the question and answer sessions for 2 questions. Any questions from the audience? And also for those who join via Zoom, you may ask by typing in the chat box, and we will check the question and we'll deliver to Kamitakahara sensei. Any questions? Maybe sensei, I have one question.
Yeah,
it's just a superficial questions, not too deep to the research itself. You said that to prepare high value-added products, then you would like to increase the hydrophilicity of the betulin, right, sensei? So what I would like to know is what do you mean about the value-added product you want?
Yeah.
Why you want to increase the hydrophilicity?
May I change to the
sorry, sensei, if I missed the information, I'm sorry.
Yeah. Yes. Maybe. Okay. So I think we have the interest on the kind of non-timber forest product, and non-timber product we have we can say the many product we have not yet used, like the betulin or the like the kind of the bark, the compounds in the bark. Wait. Maybe this slide will help your question. So not only the woody biomass, but we can adapt this concept for non-timber forest product. So as you can see, the value of the compound is highest. The pharmaceuticals, the cosmetics can be sold in higher price, but chemicals, materials in the middle part, and the fuels or energy is the lowest level for the price. So if we could convert the compound into the pharmaceutical or the cosmetics for the product, we can say that we could prepare the high value added product. For in the case of the BRIN, the bitrinic acid have very high activity. So the after preparing the such an hydro or amphiphilic compound, we can convert or we can introduce some functional group which has a very high pharmaceutical activity. That is one point how we would like to focus on in the future research. Yeah, thank you.
Thank you so much for the explanation, Sensei. Thank you so much. Any questions from the audience? Yes, Sensei, please. Microphone, please.
So thank you very much for a very kind, very interesting talk. And I'm not familiar with your research, but I have two simple questions. How did you get the raw material of the BRIN? This is first question.
Yeah, okay. Actually, for this research, I had bought the BRIN from the reagent company, but we could obtain from the bark of the white birch from the extraction process.
Okay, yeah, thank you. The second question, so finally, you synthesized the BRIN bonded with the cerobios. So what is the purpose?
Of the
BRIN with the cerobios.
Yeah. So the first we would like to confirm the reactivity of the azide group or to connect with a sugar compound. So it's very easy to analyze a low molecular weight compound by using the TLC or something. So next, we adapt the same reaction condition to the polymeric compound cellulose derivative. So that was the first purpose or the first aim to prepare the cell bios derivative. But actually, such a low molecular compound has very interesting property like the micelle formation or detergent or surface activity. So actually, we can use such a compound independently.
Okay, yeah, thank you very much.
Thank you.
Thank you very much, Sensei. Any questions, Mas Tony, from the audience in Zoom? No? Okay. Sensei, I think it's okay for your session. Please give a big round of applause to Kamitakahara Sensei.
Thank you so much.
ありがとうございます、先生。Okay, because I will be doing the same task to moderate the last sessions, I will be calling Professor Dr. Andria Augusta to deliver informations and also talk about the bioprospection of the medicinal plants of Prismatomeris SPP and also Rennelia SPP. So before that, I will be introducing Professor Andria Augusta to the audience. Professor Andria Augusta is a research professor in natural product chemistry. He is now senior researchers, professors of research in the research center for pharmaceutical ingredients and traditional medicine BRIN. And he has several research funds acquired and also several job experiences. But I would like to introduce you about the education background. Professor Andria Augusta had finished his master's and doctoral degree at Fukuyama University Hiroshima Prefecture, Japan. And he has published several journals, many journals, yeah. For examples, about together with Maehara Sensei about the anti-SARS-CoV-2 agents in Artemisia endophytic fungi and their abundance in Artemisia vulgaris tissue, and also optimization of bis anthraquinones production from endophytic fungi Diaporti sp GNBP10, and so on and so on. So without further ado and without having any delay, Professor Andria, please, the session is yours.
Okay, thank you very much, Dr. Naro. Good afternoon, everybody, ladies and gentlemen. Yeah, this is a nice strategy from committee that serve a coffee to move your concentration to foods or something. So, so I can tell everything here. Okay, thank you very much. Yeah, in this last session, I would like to discuss about biopropagation of medicinal plants that belongs to the plant genus of Renelia and Prismatomeris. Both plant genus belongs to the Rubiaceae plant family. Yeah. Okay, this is the Renelia plants. Yeah, and below is Prismatomeris. Both of plants are already used by some local people in Indonesian and other countries in Southeast Asian, yeah, for poultice of fresh wounds, and also root and bark of this plant are used for treat bronchitis, snake bites, bloody vomiting, and afterbirth medication. Also, some researchers group reported some chemical compound isolated from some species of this plant genus reported as anti-plasmodial, anti-tuberculosis, antifungal, and some compound possessing cytotoxicity activity. And also some phytochemicals also are already reported from some species of these plants that belongs to anthraquinones, iridoids, triterpene, and also phenolic compound. Okay, this, for example, Prismatomeris is also medicinal plant native from Southeast Asia. This plant distribute in some countries. Yeah, this is Myanmar, Thailand, Malaysia, Philippines also, and Indonesia. Yeah, in this map generated from JASTIP data, so many Prismatomeris species already reported from Kalimantan, Sumatra, and Malaysia, and also Thailand. And also one species reported from India here. And this is distribution of Renelia species also reported from Myanmar, Thailand, and Indonesia, mainly from Kalimantan. This is both from Malaysia and Indonesia. And more detail we can see in this map generated from JASTIP data. Yeah, just now, Professor Kamitakahara also saw this picture. This is when we went to Central Kalimantan to collect some medicinal plant sample. Yeah, at first we could get information from local people by ethnobotany interview. Yeah, we asked from local people what kind of medicinal plants they use to cure some disease, and and we try to find out the plant sample inside the forest. And also we went to Thailand to conduct the medicinal plant survey also in Thailand. Yeah, in the Thailand we can collect some Prismatomeris plant species and also Rennelia. Yeah, this is morphological Rennelia elliptica. We call in Indonesia as ginseng kuning. And this is Rennelia speciosa. Both of plant we collect from Central Kalimantan. Yeah. Some people, some local people in Central Kalimantan, they use this plant to treat cough, yeah, sometimes bronchitis. We assume that uses in the traditional knowledge probably correlated to antibacterial activity of that chemical compound contained in the plant material. So we try to check the antibacterial activity of the extracts. So we use the TLC bioautography using E. coli and Staphylococcus aureus. That our research show this spot show their antibacterial activity. And after we analyze more detail, this spot not only come from one chemical compound, but actually 3 chemical compound can isolate it from this area like this. Yeah. After isolated and purification and then analysis by a spectroscopic analysis, we can know 3 compounds, nordamercantal, damercantal, and anthraquinone derivative. 3 compound is responsible for their antibacterial activity. And also we could isolate the main anthraquinone from the acetoacetate extract of this plant is rubiadine. This is the main metabolite from Rennelia speciosa. Yeah. Actually, this anthraquinone is the same with the Rennelia elliptica. Yeah, both medicinal plant, Rennelia elliptica and Rennelia speciosa have same chemical compound in their ethyl acetate extracted from the root bark. Yeah, from the fieldwork in Thailand, we could collect one plant sample, also Rennelia speciosa. Yeah, in the plant collected from Thailand, also we could identify rubiadine as a major anthraquinone constituent. And also we could find out dimethyl rubiadine as both of compound is a major constituent in root bark of Rennelia speciosa from Thailand. Okay, we also analyze 3 species of Prismatomeris. Yeah. One species is Prismatomeris beccariana. This plant collected from East Kalimantan. And Prismatomeris tetranda and Prismatomeris griffithii both of plant collected from Thailand. From the morphological of the root bark, probably this root bark also contain anthraquinone, probably similar with Renelia species. So from tree plant, we try to make acyl acetate extractive and also later we also analyze for methanol, ethanol extractive from bark, root bark of this plant. Yeah, from HPLC profiling of the acyl acetate extract of 3 species of Prismatomeris, we could know this is Prismatomeris beccariana from Indonesia, this is Griffithii from Thailand, and this is Tetranda. 3 of plant also contain rubiadine as a major chemical constituent. Yeah, this is rubiadine, and also all sample, will contain another major constituent. Later, we isolated this compound, also belongs to the anthraquinone derivative constituent. Yeah, we conduct analysis by LCMS, and we could identify some compound containing in the acyl acetate extract of Prismatomeris, and also methanol extract of Prismatomeris. Yeah, the main chemical constituent is rubiadine, and the second chemical constituent, chemical constituent is rubiadine 1-methyl ester. Yeah, this is also some anthraquinone derivative. Yeah, but from methanol extract of the 3 all Prismatomeris species, we could show unidentified peak in the LCMS analysis. So maybe we need to identify this compound for in the future. Yeah, same with the extractive derived from Renelia species. Yeah, Prismatomeris plant also used to treat some infection-related disease like cough or others. So we also check their antimicrobial activity. So in this our experiment, most of methanol extracts of the root bar of Prismatomeris could show moderate antimicrobial activity against Staphylococcus aureus, but they show weak activity against E. coli. It means they have different sensitivity between gram-positive and gram-negative bacteria. We also check their cytotoxic activity against 2 cancer cells. This is cytotoxicity against MCF7. So from this result, we can show ethyl acetate extract from Prismatomeris beccariana. They show strongest activity compared than another 2 species. Yeah, probably ethyl acetate extract of Prismatomeris beccariana that collected from Kalimantan contain different anthraquinone derivative compared than another Prismatomeris species. Also, from the again the differential, this is G2 human life cancer cell line. Also, ethyl extractive derived from Prismatomeris beccariana collected from Kalimantan have strongest activity compared than another extract. So we assume this extract contain chemical compound that possessing strongest activity compared than another extract in this experiment. So we need to clarify what kind of compounds that are responsible for this activity. Yeah, as the result of our interview from local people also saw, inform us all of medicinal plant from Rennelia species and Prismatomeris, also in Indonesia, Malaysia, Thailand, and Myanmar. They report this plant usually used as a main tonics, to increase energy of man body. Yeah, some local people also say this medicinal plant can use as aphrodisiac. So we check that information is true or not. So we try to experiment with animal. Yeah, we use DDY mice for this experiment. Before we conduct the research to see the effectiveness of the activity as aphrodisiac of extractive, we check their toxicity against animal. So from this experiment, we could know the lethal doses of ethyl ester, not ethyl ester, ethanol extractive derived from Rennelia elliptica, bigger than 2,000 milligram per kilogram body weight. It means that extractive possessing low toxicity. Yeah, actually we need to more detail about this experiment, but we have a limited extractive from this plant. So we, at this time, we cannot increase the dose that applied to the animal. Also from the histopathology, we could see there is no abnormalities in the liver, kidney, and spleen organ of the treated animal. So after that, we conduct the experiment about the mice sexual behavior is after treatment with the Rennelia elliptica extract. We divide some groups of the DDY mice and treated with some dose of extractive derived from Rennelia elliptica. So we can see the data, but MON latency, MON's frequency, and intromission latency after treatment by extractive so not significant effects compared than negative control. And also we investigate, ejaculatory latency. It means we evaluate, ejaculation latency. It means prolongation of ejaculation latency in this and aphrodisiac action. Our data show, after treatment with 250 milligram extract for 10 days, it could prolong the ejaculation latency of the treated mice. It means increase the ejaculation latency suggests an indicator of improving sexual behavior. So from this experiment, we could get conclusion: administration of the yellow ginseng, this is the Renelia elliptica extract at those 250 milligram per kilogram body weight, could enhance the sexual behavior as an indication of aphrodisiac activity. This is our data supported the use of this medicinal plant as aphrodisiac by local people in Kalimantan and also Malaysia, Thailand, and Myanmar. Okay, thank you very much for your attention.
Thank you so much for the presentation, Professor Andria. It's very interesting information, starting from the plants, the area of collections, and also the evaluations of antibacteria, and also how say it ended. It's ended with the study of the aphrodisiac activity of mice that is stimulated by the Renelia species. Okay. Yeah, we have another 5 minutes to have a discussion. Any questions from the participants in this room? From the participants in Zoom, any questions?
Yeah, maybe already tired, everybody.
Okay. I have a question. I tried to make a question, Professor Andria.
Okay.
To be asked to you. The first is what things that triggered you to shift the research from evaluations of the bronchitis treatment to become or to find the effectivity of the substance to increase the aphrodisiac activity in mice. I mean, what finding that this is good to be evaluated its aphrodisiac activity to mice because of any trigger factors or any disease that is very how to say proficient or is commonly found in that area. I don't know. Just a question.
Okay, thank you very much for nice question. Yeah, as I said before, when we come to Kalimantan, field work in Kalimantan, Thailand, and get some information from the literature, this medicinal plant have used by local people for curing many diseases. For example, for treat bronchitis, for wound, and also they use as a tonic. Yeah, for example, maybe tonic is similar with aphrodisiac activity in the time of local people. Yeah. So we would like to know, or we would like to reveal, the local people use some kind of medicinal plant to some kind of disease. This is true or not? So we would like to get some scientific supporting for using of the medicinal plant by local people. So just now I said we got information this plant they use from coke. Yeah, we know some type of coke caused by infection or inflammation in our throat or something. And also some local people inform us this plant also used by aphrodisiac. So we would like to get scientific data to support that traditional knowledge. So we would like to reveal traditional knowledge that keep for a long time. They have a reason for it. In fact, we can support that their knowledge by scientific data. Yeah. So we should take scientific data by many experiments. I think like this.
Thank you so much for the answer, Professor Andria. Last question, last offer to the audience for asking question to Professor Andria. But if no, I think no questions from the participants join from Zoom, yeah, Pak Tony? No questions, right? Okay. Prof Andria, is it okay if we close the sessions?
Yeah, moderator.
I think Prof Andria is waiting for any other questions. Okay, before I close the sessions, to Professor, to Dr. Sopa, please enjoy the fruits. It's a local fruits. I think it's it's difficult to find outside Indonesia, especially the mangosteen, and also the citrus fruits, and also the ryogan, longan. Yeah, please enjoy the fruits. Contain vitamins to boost our immune systems for only the distinguished guests. Okay, that's all from me, and also I think this Professor Andria is the last speaker for today's JASTIP seminars. And also I would like to ask all the audience to give also not only for Professor Andria, but also for all the professors, the speakers that have been delivering their talk today. For them, please give a big round of applause again. And also, thank you. That's all from me. I will, pass back the microphone to the MC. Thank you so much.
Thank you to our speaker and our moderator. Once again, ladies and gentlemen, please give applause to all speakers and all moderators in this workshop today. Very interesting workshop. Excellencies, ladies and gentlemen, we have come to the end of our workshop agenda. To close this workshop, I would like to invite the head of Research Center for Pharmaceutical Ingredients and Traditional Medicine, Dr. Sofa Padria. Time is yours.
Dear the Honorable Guest Professor Hiroshi Kamitakahara as the leader of WP3 Just of JASTIP, and the Honorable Guest Professor Kenji Umemura as the sub-leader of WP3 of JASTIP, and the respected heads of research organization and head of in the research of organization of for health, and also their representative, and all respected professor, speakers, and JASTIP seminar participants. Good afternoon, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to express my sincere gratitude to all speakers for your time and your effort in delivering valuable topics both in poster and also maybe an oral presentation related to biodiversity. The 2023 JASTIP seminar covers various topics, genetics such as the application of high-throughput DNA, sequencing, ecology such as the application of stable isotope in ecology, and also in environment such as water treatment using tropical white rot fungi, drug discovery such as marine derived microbes, and bioprospecting innovative products such as development of a sucrose-based adhesive, and many others. This kind of seminar is very important to strengthen collaboration and to share various research results among JASTIP researchers and also for the community. JASTIP partnership is a very good example of a real implementation of multi-country collaboration. In this modern and open era world in which many challenges and environmental problems are exposed in front of our eyes, JASTIP-like cooperation is tremendously needed. In the past of 8 years, we have been sharing hits of knowledge and experiences. However, it will come to end in 2025. So we are excited to explore new opportunities for collaboration and look forward to working together to strengthen and maintain our relationship to create a better and more sustainable future. I also, for finally, like thanks to BRIN, especially for Professor Dr. Andria Augusta, and also for the all committee members who have been giving their best effort for the success of today's JASTIP seminar. Before I close this symposium, maybe I give the pantun. A monkey is stealing apple pie. It presses a button to open the gateway. I promise this is not a goodbye. I will see you again someday. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Sofa Fajria, for closing this event. Ladies and gentlemen, allow me to read you a pantun. Buah nanas, buah ceri, dicampur gula enak sekali. Cukup sekian hari ini, semoga bertemu di lain hari. Sungguh manis buah ini, buahnya dimakan dengan cepat. Berarti sudah acara ini, semoga bisa memberi manfaat. Once again, thank you very much to all participants for your active participant. I am Berlian Irawan. I will say goodbye and see you again. Wassalamualaikum warahmatullahi wabarakatuh. Iya, diinformasikan kepada seluruh peserta hadirin yang belum mendapatkan goodie bag bisa mengambil di meja registrasi ada di depan. Terima kasih. Dan yang belum menikmati coffee break, silakan untuk menikmati coffee break-nya ada di luar. Terima kasih.